So thank you all for coming. Let me say a few words about the event. As many of you know, we have these symposia 3 times a semester or so. And somewhere during the pandemic, we weren't the only ones, but we had the idea of devoting at least one event to younger speakers and postdocs. It was a pandemic, so we did it online, and it was tremendous fun. We heard about an even broader, over the course of the semester, we tend to cover many different topics, but here we were able to do it in one day. So it kind of keeps you on your toes. You try to follow things far from what you perhaps usually listen to, which is good for all of us. The other thing I have to say is the talks were just incredibly good. So we decided to keep doing it. And it's an opportunity to hear from a much wider range of people from all over the world. Thank you for thanks to all of our speakers for coming. It's fun to visit New York, but it also is a project to get here on short notice. We keep trying to organize ourselves so you get more notice. It doesn't always work. And the talks are grouped roughly by themes, so there'll be 3 sets of talks. The grouping is approximate, so be prepared for that. Our standard in these events is to do things in the physics style, so polite interruption is encouraged. Speakers have the right to say, no, I'll talk about that later. Our advice to the speakers was you have half an hour and you should prepare 20 minutes on the assumption that the audience will take care of the rest. And the only thing we don't have in the room is a clock. So I might have to service the clock. I don't know. We'll see what we can do.
Hopefully I'll be in the back. But I'll give.
And actually each speaker can decide how they want to do it. So let's get the day started. Fotios is coming to us from Amsterdam and is going to talk about signaling.
So yeah, thanks for the introduction, Bill. I am a graduate student working with Hopkins over at AMOLF in Amsterdam. And this is a project I've been working on for almost 5 years, and it connects chemotaxis, criticality, and giant signaling arrays. So 2-state systems are very prominent in biology. The textbook example of a 2-state system is the ion channel, and I literally took this picture from a textbook. Of course, in the ion channel, you have a system that switches between an open and closed conformation to either let or block ions. And another typical example of a 2-state system is the bacterial motor. There, this is the E. coli motor specifically, which switches between a counterclockwise and clockwise motion very fast within a few milliseconds. What's common between these 2 systems, it's their size. They're both very small. The sodium channel is comprised of something like 6 or 10 proteins, and the bacterial motor is closer to 30. We, for the first time, have seen stochastic 2-state switching in a much larger system, in the whole E. coli chemoreceptor array. And this is fascinating because the size of the system is order of magnitude larger. It's something like between 1,000 and 10,000 chemoreceptor units, all of them well ordered in a hexagonal lattice. But before I tell you more about what we see, I will tell you more about what we measure. We're using a FRET assay, a fluorescent resonance density transfer assay, that we've developed in our lab recently. So the chemotaxis system is a sensing system, and of course its main part are the sensors. E. coli has 5 different chemoreceptors. They sense almost all 20 amino acids, sugars, dipeptides, but also physical cues like temperature and pH. These chemoreceptors are organized in a lattice, and the signal is integrated by a kinase called Ka. And this kinase, the brain of the cell if you wish, phosphorylates another small protein, Ky. Ky is a response regulator. It's small, it diffuses fast, it binds to the motor, and when it's phosphorylated, it induces a tumble motion, a clockwise motion of the motor, so a tumble motion of the cell. And then there is another small protein, KZ. KZ basically tries to do the action of Ka. Ka phosphorylates Ky, KZ dephosphorylates Ky. So when there is no ligand bound to the receptors, Ka is active and the cell tumbles, so it can find a new source of ligand, a new source of food. On the other hand, when there is ligand bound, Ka is inactive, Ky is not phosphorylated, it doesn't bind to the motor, so the cell runs. It runs towards the source of the ligand. And what we've done now is to fuse genetically Ky to RFP, a small fluorophore, and Kz to another fluorophore, YFP. And when the two come in close proximity, which is when Ka is active, we get a fluorescent signal through resonance energy transfer between the two fluorophores. And this allows us, at a single cell resolution, to measure Ka activity. So using this system, this is a typical picture of the cell we see, right? These are all single cell trajectories. We have a good signal to noise ratio. Usually the cell is biased at either the one state, the active state, or the inactive state of the kinase. And as soon as you give attractant to the cell, the kinase shuts off. Of course, if it's already off, it stays at off state. On the other hand, if you add repellent, it goes back to 1. If it's at the 0 state, it's now strongly biased towards the active state. But when we did this experiment for the first time, we've noticed something very strange. Many cells, something between 10 to 20% of the cells, switch stochastically between the on and the off state. This is without any ligand. These cells are in buffer, they're temperature controlled, and yet they switch, they do these giant fluctuations between the two states. And this is true for the two major chemoreceptors of E. coli, both TAR and TSR. TAR and TSR, I should say, it's something like 95% of all the chemoreceptors. In this case, we would express a homogeneous population, which is either TAR or TSR. And these cells are still chemotactically active, right? If you add ligand, if you add attractant, the activity will go to 0. If you add repellent, the activity will go to 1. So, of course, the first thing we thought was that maybe they're actually sensing something. Maybe there is something in our buffer, something in the environment, E. coli can even sense light, that causes these fluctuations. So to check that, we genetically broke these receptors. The receptors are still there, but they cannot bind any ligand. And you can see that here, if we hit the cells with different kinds of ligand, there is no response. This is the average response. If you look at individual cells, they still switch, right? So this is a very good indication that the switching we see is stochastic rather than sensing some kind of unknown ligand. And then we want to take a step further and analyze those fluctuations, those switches. To do that, we wrote code that automatically. Yeah.
Why is it that why you only touch each other when you want to?
So it does, but at a very low rate. I think there have been experiments. Exactly, it falls off very fast. Yeah, there have been experiments, I think, by Victor Surgic in the early 2000s, and it shows that it falls off very fast. Exactly. Yeah. So yeah, it's quite hard to analyze those, right? Because they don't sense ligand. It's impossible to normalize those time series, right? You don't know if these are full-scale switches or not. You don't know if this. It's impossible to know. It's impossible to know if this is 0 or not, right? Because if you don't sense ligand, you don't have a 0, right? Also, you don't have a 1. So we haven't really exact statistics from those cells because there is no good way to analyze. Of course, you can argue that you can use the noise, right, as a proxy for the amplitude, but this is not very clean. So. Yeah, by eye, you clearly see it, right? You can see it here. Yeah, so there's clearly switching in the absence of ligand binding, which is proof that, switching something intrinsic to the cell rather than sensing something that we haven't accounted for. So then we wrote code to extract those switching statistics. And we extract two things mostly. The transition time. This is the time it takes to go from the up state to the down state. This is tau minus. or the transition time down. And then we extract the transition time from the low state to the high state. This is tau plus. And we also extract the residence time. This is delta t. Delta t down for the time the cell spends at the 0 level, delta t up for the time the cell spends at the level 1 at the full activity level. Using this, and after acquiring thousands data from thousands of cells, we can extract distributions. And we see that the residence time distributions, the delta t time distributions, are exponentially distributed. So if you're at 0.5% bias, activity bias, so if the cell mostly spends equal amount of time up and down, of course you see that the delta t up and delta t down distributions are identical. If the cell spends most of its time on the high state, so if the activity bias is high, the distribution for delta t up, of course, has a longer tail, right? And conversely, if the cell spends most of its time at the low activity state, delta t down, the blue here, has a longer tail. And this is true for both TAR and TSR. They look very similar. Next, we can take the average residence time, both the up and the down average time, and plot that as a function of the activity bias. And if we take the log of these two quantities, we see that they scale approximately linearly. And you can take the activity bias as a proxy for the free energy barrier difference between two states, right? So this is an indication that this might be just a simple barrier crossing process that requires no energy. And we can also extract the characteristic time scale of those transitions, tau, which is roughly 50 seconds, roughly a minute. Oh, no. So indeed, you can set it. You can set it with ligand. We don't do that. But cells by themselves, they have an activity bias, which we don't know exactly how it's determined. Maybe it's determined genetically. You can also change it using temperature, but we fix the temperature. Exactly. So we measure the activity bias, and we define it as the time it spends at the up state and the low state. So if you take 1 over that, this is the activity bias. Yeah. Yeah. No, this can sense, because again, we need to normalize. So at the end of each experiment, we hit it with ligand to get the 0 and the 1, right? Which is how we get the activity bias. Yeah. And then we can also extract the transition times. And we can plot the distribution, and we see that these are very slow transitions. The bacterial motor I showed you earlier switches within milliseconds, the same with the ion channel. These switches take more like 5 to 6 seconds. So they're unprecedentedly slow, which might be an indication of the very large number of cooperative units that are involved in the system, right? I just told you that there are like 1,000 or 10,000 receptors in those cells. And again, TSR and TAR look very similar regarding the statistics. And something that's interesting is that this transition time does not depend on the activity bias. No matter whether the cell spends more time on the high state or the low state, the transition time is always 5 to 6 seconds, both for TAR and TSR. And next, we wanted to see if we can simulate this behavior. And perhaps the most famous lattice model is the Ising lattice. I'm sure that all of you are familiar with the Ising system, but I will quickly explain it. Its Ising lattice has 3 important parameters. Here we're only simulating square lattices. So one important parameter is L. This is the linear dimension of the lattice. All our lattices are L by L, so the number of units is L squared. And the next important parameter is J. J is the coupling energy between two neighboring spins. There is the J of the system, and there is also a critical J, J star, which is determined by the size of the system. And there is an analytical solution for that, for this J star. If your lattice has an energy lower than the critical coupling energy, J smaller than J star, the system is unpolarized. It stays at the 0.5% activity or the 50% activity, and then it just fluctuates around the mean. If now you have a coupling energy that's high, much higher than the critical coupling energy, the system will become biased. It will either be at the 0 or the 1 state, but it will stay there. You never see switching between the states because it's a very rare event. The time you need to wait, it's close to infinity, depending on, of course, on the coupling energy. But finally, there is a state between these two extremes where J is roughly equal to J star. where J is roughly equal to the coupling, the critical coupling energy. And there you see switching. You see stochastic switching between 1 and 0 and 0 and 1. And perhaps you can already tell that this looks very similar to our experimental data, right? And of course, this got us very excited. And we tried to analyze the stochastic switches, the sizing switches, using the same method, the same code. And there you see that if you extract the residence time, delta t, either up or down, you see that the distributions look almost identical to the distributions we get from experiments, right? You have the same exponential distribution. And if your system is biased at the high state, and now we do bias the system, we have an external field, so we can either turn the field to a positive value, which will bias the system to the up state, or to a negative value, which will bias the system to the 0 state. We see that the scaling is exactly the same as in our experiments. If you're mostly in the high state, delta t up has a longer tail here. Next, we also extracted the transition times, and this is quite remarkable. The transition times have exactly the same shape, the same peak as in the experimental data. This is not a fit. We just took a lattice that had approximately the same switching statistics as our experiments, and we plot the data. And you see there is an excellent agreement between experiments and data. Exactly. That's very true. I will get back to that later. But yeah, we simulated many sizes basically. Which maybe no, it's the next slide. So okay, before I go to the next slide, I will quickly say that again, the bias, the activity bias, which we now tune using an external field, does not affect the mean or the shape of distribution. It's always the same shape, the same mean, no matter the activity bias. And indeed, we simulated many pairs of L's or N's and J's. Its color here is a different energy, a different J, and the x-axis is a different linear dimension of its Ising array, Ising lattice. And what we do to compare simulations and experiments is to extract the ratio between delta t and tau. This is a dimensionless quantity, so we can easily compare experiments and simulations. And if you extract this ratio for the cells, you get a ratio either close to 8 for TAR or closer to 12 for TSR. And the ratio of 8 means that the transition time is 8 times faster than the residence time, right? The residence time is about a minute. The transition time is closer to 5 or 6 seconds. And as you can see, there are many simulated pairs of N and J that have the same delta t over tau as in our experiments. These are the points that overlap with those lines. And if you plot the isoline for those points as a function of J and L, you see that you're very close to a regime between the polarized and the non-polarized state. So here with blue, we show pairs of J and L that are non-polarized, so the blue line here. And in this regime, we're polarized, right? These are pairs of J and L that contribute to a polarized lattice. There is a boundary between the two. This is the gold line, and this is the critical energy, J star, for its combination of J and L. And what you can see is that both TAR, that's a ratio of 8, and both TSR, that's a ratio of 12, are very close to this critical point. They're very close to this golden line. And if you rescale J by J star, you get a straight line at 1, this critical point. You see that both TAR and TSR are remarkably close to this critical point. TAR is slightly below it, TSR is slightly above it, but they're both within 3% of the critical point. And this is amazing. This is amazing because it tells us that the whole bacterial chemotaxis system, the whole receptor array, all these thousands of receptor units are poised very close to the critical point. And this is very relevant, right? Lately, there's been a shift, I would say, in cell biology, where we started considering first-order phase transitions. First-order phase transitions appear everywhere, apparently, in biology. It appears in structuring, multi-phase structuring without any need for a membrane. It appears in cellular function, in transcription translation. It also appears in metastability, where we can get a liquid and turn it into a glass or a gel using a first-order transition. But now we've seen a second-order transition. Unlike the first-order transition, second-order transition has a critical point, right? It has a point where you can switch between an order and a disordered state. And our receptors, this massive lattices are poised close to criticality. A natural question, a natural follow-up question is how fast do those spins, those arrays switch, right? We have a transition time, which is the time to go from the active to inactive state, but this is for the whole array. The question is, what happens for a single spin in our system? And there is no easy answer because we don't exactly know what's a spin in this case, right? But we can theorize. The smallest unit that it has been shown in vitro to have sensing properties is the core unit, which is a Ka dimer to Kw monomers, and Kw is a scaffolding protein. It's a protein that keeps everything together, basically. And 6 trimers of chemoreceptor dimers. Chemoreceptors always form dimers, and in this core unit, you have 6 trimers of those dimers. On the other hand, if you look at the structure of the lattice, and we know the structure thanks to amazing cryo-EM data, you can see that there is a repeating unit cell, right? There is a structure that repeats in the lattice. And this repeating unit cell is comprised of a full core unit and then 4 half core units. I hope this is visible, but you can see this unit cell here bound by this box. So this can constrain our L, right? The L of our chemoreceptor array. And then if you take the size of this L and then you divide by the residence time in our experiments, we can get an approximate time, an approximate range for this omega, this switching frequency of the system, of a single spin in our system, which we found to be between 15 and 40 milliseconds. This is really cool because you cannot extract this thing in vivo. People have extracted these switching times in vitro using NMR, but it's the first time somebody was able to extract that in living cells. And then a natural question is, why criticality, right? We care about criticality because we're physicists, but why should the cell care about criticality? Why is criticality something useful for the cell? And the answer actually is quite simple. So here we plot the response speed, the time it takes for the array to switch, as a function of the response amplitude, which is a measure of amplification, of course, a measure of gain. If you have very weak coupling, as you go closer to the zero coupling limit, you see that the response is very high, the spin of the response is very high, but the amplitude is very small. So even though spins can switch quickly between the up and down state, there is no amplification. On the other hand, if you make the coupling very strong, this is close to the MWC limit, where J approaches infinity, you see that the amplitude is very high. You have very high gain. You can switch your whole array very easily, but the speed is very slow. It takes time, of course, to switch a massive array of spins. And now, if you're sitting near a critical point, you get the best of both worlds. You're between the no coupling limit and the infinite limit, which means that you have high response amplitude, but at the same time, you also have high response speed. And it turns out that if you plot our data within this range, we see that all our experimental data fall very close to this critical point. So you're at the sweet spot, basically. You can respond fast to ligand, but you can also respond with high amplification, which both are very important objectives if you're a cell. And normally this would be the end of my presentation, but since I'm here with many excellent physicists, I have a question for you. So I think I've shown earlier, but I didn't talk about, is this. There is a slight asymmetry between up and down transitions. In fact, going to the down state is always slower by perhaps 1 or 2 seconds than going to the up state, going from down to up. And this is true for both TAR and TSR. Of course, this gets lost if you rescale by the mean, which what I've done here. But on the other hand, if you look at the transition times in our Ising simulations, there is no asymmetry, right? Up and down transitions have the exact same shape, the exact same mean, which is not true for experiments. Of course, both don't depend on the activity bias, but there is a clear difference regarding the asymmetry. And of course, before you ask, I should say that this is not an effect of our analysis. We use the same analysis code for experiments and simulations, and we don't see the asymmetry in the simulations. And we've also gone a step further. We've simulated experimental time series. We've injected noise. We've injected noise to match the noise we get in our typical experiment. This is white noise. And if we load a time series without an asymmetry between up and down in our code, we don't see an asymmetric distribution. So we're fairly sure that the symmetry we see is real. So this is my question to you. Our Ising model, of course, it's an equilibrium Ising model. It obeys the tail balance. It doesn't break the tail balance. And we don't see an asymmetry there. So my question is, can you somehow tune an equilibrium model to see this asymmetry? And if no, is that a signature for non-equilibrium driving? That's an open question. We can also discuss it later. And this brings me to the real end of my presentation. Quick summary. We've seen stochastic two-state switching of entire massive chemoreceptor arrays using single-cell FRET. Switching is observed in the absence of any ligand or methylation feedback. If you break ligand binding, you still see switching. The Ising model simulations reveal a near critical tune of cooperativity. For the first time, we can show that a signaling system in vivo employs a second-order phase transition to tune itself, to pose itself close to a critical point. This near critical tune balances two performance objectives: high gain, high amplification, and fast response to ligand, which is something a cell, of course, wants. And finally, we see an asymmetry between up and down transitions, which so far is the only feature that our Ising model cannot explain. And before I end, I want to quickly thank those people: Tom for the supervision of these years, Johannes, a previous graduate student of Tom. We did all this analysis together. Simone was our group technician who made these mutants, and Sandy Parkinson, a legend of genetics, who designed those activity mutants. And finally, I would like to thank you for your attention. Thanks.
Coupling between the attractor-network and in particular, I'm curious whether you think it's susceptible to kinetic or environmental versus.
Excellent question. Excellent question. So the answer is yes. I do think that it's susceptible to kinetic manipulation. So we don't exactly know how it works, how the coupling works, but we think, yeah, sorry, it's here. We think that the scaffolding protein KW is involved, right? KW has no clear function. It's there to keep everything together. But turns out that our collaborators have data that show that if you overexpress KW above the native level, you get higher cooperativity between receptors, but not higher sensitivity. So I wouldn't be surprised if KW is somehow involved in this critical tuning, if KW is what determines this J between receptors. And we're currently trying to do experiments to prove that. But yeah, it's a very good question. Thanks. Please go ahead. Yeah. Yeah. We have, yes. So this is a very good point, something we thought some time ago. So it's true that dephosphorylation have slightly different time scales. And the sign is correct. The sign would explain what we see. But the difference is in the order of milliseconds. People have measured that. And our difference is the order of seconds. And to further show that the symmetry we see, it is overexpressed. Yes, but what we've done was to hit the cells with ligand and image very quickly. And there, of course, we still get a transition that is dictated by ligand. And then the up and down transition is we cannot see a difference with our acquisition frame rate, which is a tenth of a second. So I do believe that what you say is true, that there is a symmetry there, but I think this asymmetry is much, much slower than what we see. And further proof is that we've lowered the KY/KZ expression to match the native levels, and we still see an asymmetry. We still see switching, and we still see an asymmetry. Yeah, thanks.
I thought if I take the Ising model and I point to the critical point, if I look at the distribution of magnetization,
yeah,
I see something that does not have to be, but rather is flat on top.
Yeah.
So if I think about the dynamics that equilibrium statement, so now you have to do kinetic.
Mhm.
But that doesn't come from the whole thing switching, right? It comes from kind of giant fluctuations. So I guess I'm surprised that you see something that looks like switching between the two fully magnetized, the fully up and fully down state when you're employed for the
I agree. It is a surprising result. Unfortunately,
I'm also surprised you see that in the Isaacson.
we are surprised too. It wasn't a clear expectation exactly, and I don't have a good intuition because I used to believe exactly what you said. So I don't have a good intuition on why you see switching, and yet you see full scale switching. Yep. Any other question, perhaps?
That dotted line is like all the lines is plotted, they are different types.
Yeah.
Why the dotted line is plot, okay, on side.
Perhaps I should switch to the other slide. Yeah, these different colors. So you're talking about this line, right? This line in the middle, yeah.
Low temperature phase. And since you are in the low temperature phase and the size of the system is small,
Mhm.
you're like hopping from one phase to the other.
Mhm.
So you must be in the low temperature phase. If you find that to measure the susceptibility of the system and you like different values of the different curves, you will see the susceptibility increase. Because like all these curves are in the low temperature phase.
Right. Yeah.
So the reason why you see this hopping from one to the other is because of that.
Because you are close to the low temperature limit. But you still see switching if you go above the critical phase, right? This whole regime here is still switching. No, this whole regime here is still switching.
Yeah, but that's like
But above the critical point. Oh, so you say the switching doesn't have to do with criticality necessarily, but it has to do with the temperature limit.
yeah, you're like
Yeah. Right. But then should you expect to see a difference whether you are above or below the critical point?
yeah, you're below. It's like huge fluctuations without switching.
No, I mean, if you're close, so in both cases, we're close to the critical point. If you're below or above, do you expect to see a difference in the switching behavior?
You're during the high temperature phase, you are not
Yeah.
something that is fluctuating along. So basically, the black curve in the plot will become bigger and bigger.
Yeah.
At some point, you see like this point.
Yeah, no, that's true. That's true for sure. Yeah, yeah. That's interesting. But maybe we can discuss it further after that. Yeah, that's an interesting point. Yeah, thanks. Thank you.
Thank you, the organizers, for this opportunity to share today this work that I've been working on in the laboratory of Gonzalez at UCSD. So we had a very general question, and it's how dormant cells process information. So let me start from the beginning. So in the lab, we work with Bacillus subtilis, that is a bacteria that, as many other microorganisms, when they are stressed, they form these bacterial spores that it contains this coat, and inside they have the genetic material. So when they are stressed, these spores are formed, and they are dehydrated. So and these spores are dormant. And what I mean by dormant is that they don't do any metabolic activity. They are just almost like dead. So some people consider them that are almost like rocks. So, and they are so dormant and resilient that some labs have found spores in amber, and they can be alive for millions of years. There are some groups that also send them out of space, and they can survive. They are a major source of antibiotic resistance, and they are also a major source of food contamination. So as you can imagine, it's very important to understand how these bacterial spores work. And there are many labs that work with bacterial spores. There is a lot of known about the formation of the spore, that it's known as sporulation. So when there is this stress, the bacteria form this spore inside, and then the mother cell lyses, and then we have this spore. And there are also a lot of experiments about the germination process, that is when the spore gets back to life again. And usually they do it when they are in the presence of some nutrients that are called germinants. So this is the process of germination. But very little is known about this moment when the cells are considered like rocks, how they process information. Because in one side, they need to be very robust. They cannot germinate in the first stimulus that they receive, but they need to be sensitive enough to germinate when the environment is correct. So we wonder like how these dormant cells are processing the information. So to do that, we have this experimental setup where we have the microfluidic system. We can flow media on and off, and we have this cell trap where we can contain several thousands of spores. And here in this movie, I'm going to show you how a bacterial spore germinates. So if you remember, I explained you that these are dehydrated. So during the germination process, you will see that they become black because they are internalizing water. So there is this the hydration of the spore. And in this experiment, we have the addition of germinant that it will be in time equals 0. So this is what we observe. And as you can see, this is a very short process that they just enter water inside of the spore and then are germinated. But spores are not in a constant environment. They are usually found in the soil and things are changing over time. So we wanted to know like what's happening when we are doing these pulses of 3 minutes just because of a technical limitation. And then we were waiting 2 hours between pulses just to allow all the spores to germinate from the pulse of germinant that we are giving to them. And here I'm going to show you now several spores. And in this plot, we will see different lines, and each line will be one spore. And it will go from light gray when it's dormant, when it has the white phase, to a darker color because it will be the germination. And this will be over time. So let's see what happens. Each line is when we are hitting with this germinant. So this is at the end, we have all the spores germinated. And we can quantify this. For example, here I'm showing the dormant fraction of the spores. So at the beginning, we have that 100% of the spores are dormant. And then in the first pulse, we have like some spores germinated. And I don't know if you pay attention, but in the second pulse, there were much more spores germinating. We can also quantify this in a different way. We can show the germination probability. So we have that in the first pulse, there were some spores germinating. And in the case that there was no any kind of processing of information, we would expect that the germination probability should be also the same than the first pulse. But in this case, we are seeing that in the second pulse, more spores are germinating. So this should mean that there is some integration capacity of the spores. They have seen that there are different pulses, so they are integrating this information. They are trapped in a chamber. So yeah. So they cannot move around. So as I said, they are completely dormant. They don't have metabolic activity. They are not producing any kind of protein. So how can a spore integrate this information? So one of the things that I didn't say is that spores are packed with ions. And here I'm going to skip a lot of context, but in the lab we have been working with potassium, and we know that potassium is very important for any cellular function, and is the most abundant ion in any living cell. So we thought that maybe potassium was playing a role in here. And we were inspired by the integrate and fire model that neuroscience use, where we have the spore, and we have the extracellular potassium that is defined with by this equation, where we have the conductance of the potassium, and also some in a specific conductance from other ion channels. And then we have the relaxation term, and then we have also the intracellular potassium and also the voltage of the cell. So with that, we have that the initial potassium content and also the conductance of the potassium dictates the dynamics of the potassium content in the spore. And this will give us the germination propensity, how easy it is for the spores to germinate. Let me explain in a different way. So we have the germination propensity. The cells, the spores, when they are formed, they have some intracellular potassium from the beginning, and this is what determines how far they are from the germination threshold. Every time that the spore is exposed to a germinant, they will efflux some potassium. So this will help them to move towards the germination threshold. And once they reach this germination threshold, they will fire the germination. Okay, so this is what we get from the experiments, and this is what the model reproduces. So it seems to be in agreement. But like, it seems that instead of these spores just sitting there, being like rocks, they can work as capacitors. So during the sporulation process, they will build up all this potassium in the interior that later they can use to do some work. In this case, it will be to move towards the germination threshold and then germinate. So now we wanted to test this model. So there are two main keys that we need to test. One is modifying the initial potassium concentration, and the other one is to modify the potassium efflux. So for this talk, I'm going to center into these two particular experiments. So the first one is changing the initial potassium concentration. So for that, what we did is it was to knock out the importer of potassium. So during the sporulation process, these spores will have a harder time to internalize this potassium. So what we are expecting is that instead of like wild type spores will be residing here, but for these mutants, they will be closer to the germination threshold because they have less potassium in the interior. So I'm going to show you here a movie where we have wild type in the top that we saw already previously. So let's put our attention into this mutant, the KTRC, and we can see that already in the second pulse, almost all spores germinated compared to wild type. So they are more sensitive to the germinant. And this is just because they are closer to this threshold. So we have the experiments, and then it seems that the model it's reproducing the same kind of results. And we can, again, measure the germination probability. We see that these KTRC mutants have a more probability of germination. But when we measure the integration capacity, we see that it's exactly the same, because in here we are not changing how easily they approach to this threshold. The only thing that we are changing is how close they are to this threshold. So the integration capacity is not affected because it will be due to the potassium efflux. And then the second experiment, it's with modifying the potassium efflux. And for that, we used a quinine, that it's a drug that blocks the potassium channel. So in this experiment, we will have that all the spores will reside from the beginning in the same position than wild type. But what we are doing is modifying how easy they approach to this threshold. And here again, there is a movie, top wild type, bottom with a drug. And now we can see that these spores have a harder time to germinate. So they need more inputs from the germinant to reach the threshold. And this is because we are changing in this case how potassium efflux. Again, we have the experiments and the model in agreement, and we have that there is less germination probability for these spores, but then and also we have that the integration capacity in this case is affected because we are changing the efflux of this potassium. We also measured the extracellular potassium at the single cell level, because if we are thinking that every time that we hit with the germinants, there is this efflux of potassium, we should be able to see this potassium in the media. So for that, we are using an extracellular potassium indicator that it's called APG4. And then in here, we are seeing at the single cell level how this potassium efflux every time that we are hitting with the germinant. But in the case of the quinine addition, that is the blocker of the potassium channel, we cannot see these changes in the extracellular potassium. So I think that from here, we have established that spores are releasing this potassium every time that they see a germinant exposure, and they use this to know how far they are from the threshold. But we wanted to go a little bit further. And because they are releasing this potassium, the model says that, of course, they are changing their membrane potential. They are becoming more and more negative because they are releasing all these positively charged ions. So we wanted to visualize this at the single cell level also. And for that, we used thioflavin T, that is a positively charged fluorescent dye. And this dye will stick to the coat of the spore, and we will see it brighter and brighter as they become more negative. So here I'm going to show you a movie where we have a spore with the fluorescent dye, and also here in phase the same spore. And in this plot, I'm going to show you the electrochemical potential that it will be measured by the fluorescence of THT and over time. So in here, we can see that every time that we are hitting this spore, there is this change in the electrochemical potential. But most importantly, this cell still remains white, so it still is dormant. We are not, so the spore is not doing anything from the outside, but in the interior, there is all these changes in the membrane potential. So to sum up, what we have seen is that spores are dormant cells that can integrate information by releasing these potassium channels, this potassium to the exterior every time that they encounter the germinant. And they use this to do the work of approaching to the germination threshold, that then it will fire the germination process. And this work has been published last year. So I encourage you to look at it and find more the details of this project. And with that, I would like to thank the other co-authors of the paper, especially Kaito Kikuchi, who is also first author, and Colin and Professor Jordi García-Ojalvo, who were the ones that developed the mathematical model. And with that, I would like also to thank you for your attention. Yeah.
So it seems like there is this cleansing effect where the like almost a fluorescence thing, where the bacteria, like when everything's releasing potassium, in that sense, like the total amount. So have you tried like diluting just the number of cells and seeing how if that would change?
Yeah, we haven't seen a correlation. So we are in a microfluidic system, so there is always flow. So I'm assuming that like all the potassium that they are releasing, it will be flow away. So maybe in parts, if we don't have this kind of flow, maybe we could see some effect, because we know that we can clamp the potassium efflux. So if you have the spore and you add a lot of potassium in the exterior, the potassium cannot go out. So maybe if you have a lot of spores and they are releasing this potassium, this could affect to the neighbors.
Right. Yeah.
But because we have the microfluidic system, we cannot see it. But it would be interesting to, like, in another setup, see what happens. Yes. Yeah. Yeah.
I wonder if there was a nutrient dependence in its decision making, like for instance, whether it contains a longer memory, if it has a higher quality nutrient or something.
So let me see. So I don't know if this is exactly what you are asking. So it depends on the amount of nutrients that we have, but in the first, in the first pulse, it doesn't depend that much. It only affects on the second pulse, the amount of nutrient that you have. Yes, and about the memory, so yeah, we have, so yeah, I think that it will affect on the second pulse, but not in the first. Yes, yeah. I don't know if I answered. Yeah. Yeah. So spores, there is a little bit of debate in the field, but spores are almost depleted of ATP. So that's the key question, like how can these spores do any work if they don't have ATP? How can they integrate information if they don't have the ATP to use it? And this is just a free energy process using the potassium release. So there is no ATP involved in this processing. Yes.
So it seems that unlike with neurons, the relaxation time is very slow.
Yeah.
So my first question is, does that make sense in light of the biological mechanisms?
So as I said, that these spores can be there for millions of years. So if you have some nutrient that it's not enough for make them germinate, they should forget it, because like if they see another pulse, like I don't know, like 1,000 years later, this cannot be accumulated. So there should be. They forget it. So this is not included in this work, but we have continued working, and it seems that it's around like 20 hours or so. So there are some hours, which in a scale of the years is not that long.
My next question was, have you checked if your model, which I assume has like some parameters like the relaxation scale, does that generalize to different time courses of pulses?
Can you repeat it again?
Yeah, so you have like a fixed model,
Yeah.
which has a certain relaxation time, etc. Have you tried varying the time at which the nutrient pulses come in? Like instead of separated by 3 minutes, it's separated by 6 minutes, and seeing if the model generalizes?
Yeah. We have done that, and the only thing is that we don't know how the cell recovers. So this is for this reason we are working on that. But yeah, so the model could do it, but we don't know what's the mechanism of recovering. I don't know if I answered your question. More or less.
I'm not sure what recovery actually context will work. Back to baseline.
Yeah, going back, like if they germinated like a certain amount in the first pulse, and in the second pulse we usually see that a lot more germinate. Like if we see then the same amount than the first pulse, this is for me recovery. I don't know if we were talking then about the same kind of recovery. We can discuss later if you want. Okay. Yeah, if we add potassium in there, we are using L-alanine as a germinant. And if we add a lot of potassium in the outside, it quenches the germination, because potassium cannot go outside. There is this equilibrium, and then they cannot flow. Yeah.
I just have a basic question. How the dynamics would change if instead of increase in the internal potassium population decrease it at a baseline?
So in the interior of the cell. Yeah. So we have this mutant, the KTRC, that it reduces the amount of potassium inside. And then we see that they are more sensitive. They germinate faster with less stimulus. And we have also the experiment where we charge the spores with a lot of potassium inside during the sporulation process, and we see the opposite, that now they need more input to germinate.
How do the dynamics change in the increased potassium? I mean, how do they compare to the increased potassium and the decreased A rate, the GK increased dynamics?
It doesn't affect the GK.
Oh,
right?
I meant in terms of the GK dynamics, how does it compare to when you have like a higher internal potassium?
When you have higher internal potassium, they are farther from the threshold.
Yeah.
So they need more time to germinate, right?
Yeah.
I don't know. Is this not the answer?
Oh, yeah.
Oh, okay, good. Thank you.
So is your picture of recovery that they're actually replenishing intracellular potassium?
This is what we don't know. Yeah, this is. Yeah.
I mean, you could test that, of course.
Yeah. We have
potassium would make it easier.
Yeah, that would be the easiest, but it should go against the concentration. So
Yeah, well, I was going to suggest that you check if they have those potassium.
Bacillus subtilis doesn't have potassium antiporter. Yeah.
So then you gotta try to get energy source, right?
Yeah, that's the problem because they need to uptake the potassium inside, but this would cause ATP, and spores don't have ATP.
Yeah.
So we are still in the puzzle that we don't know how they recover. It cannot be potassium because it goes against. Yeah, and you're suggesting that maybe it's sodium, but they don't have antiporters for Bacillus subtilis. Yeah.
So how does, because I can remember in potential things really increase a lot.
Mm-hmm.
Like how does this compare to the actual normal operating membrane potential of the bacillus? That would be also like just leaving the membrane potential.
Wait, that would arbitrary unit. Your membrane potential.
Yeah, for the THT, yes, that's arbitrary unit. But no, yeah, but but it's true that we have. Yeah.
Actually, I have a related
Yeah.
question.
Okay.
There are
Yeah.
dyes that insert themselves in the membrane
Mhm.
and change their effector on the membrane.
Yeah.
So I don't actually know whether these are bright enough to use in single bacteria or not.
Yeah. Yeah, we haven't tried. We tried TMRM, that it's another fluorescent dye similar to THT that doesn't change in color. What it does, it's only that it binds if it's more negative. In that one, we have exactly the same data. I can show you if you want. But yeah, you mean like some ratiometric dye?
I mean, membrane potentials, if you translate it to electric field, right, they're very large.
Mm-hmm.
So if you put an organic pigment in the membrane, the Stark shift is enough to lose the spectrum, not to see it.
Mm-hmm.
Typically,
Yeah.
There was a period where people did lots of experiments for different reasons, some in bioenergetics, some in
Yeah, we could try. Yeah, that's a good. We never tried that, and yeah, thank you. Yeah.
So going back to the first question, obviously this idea of could they be sensing other things in the environment if you're watching it all the way, sure. I guess can you give me more context for where, like when they're forming an amber or whatever, do you find 5 spores? Do you find 5,000? Like what sort of dynamics might they have in their physical environment? Because in some way, are they almost like it's an inhibitory form center where one can germinate and spread out and all of their neighbors would be
I see. So let me break a little bit your question. So usually spores are formed in biofilms, and there are like thousands of spores together packed in a matrix environment. And then what was the other what was the other part of the question? I don't remember now.
Yeah, so just thinking about like obviously in flow we're gonna wash away but would you see a case where it is a biofilm and then a few of them are able to germinate and escape growing and the rest are all suppressed?
Yeah, so in that there is a paper that they have seen differences in the germination if they have like a large amount of spores or very few. But yeah, again, like we don't know because we were doing the flow. But yeah, it would be interesting to see it. Yeah, especially because we see that potassium affects the germination, and if they are releasing potassium, they should affect the neighbors. Yeah, totally. Yeah.
This might be a really dumb question, but can you do the same experiment by just like completely like applying some kind of electric field to completely change like the electronic environment, so then that controls what the potassium goes in or out?
Yeah, we are working. Yeah, we have now electric device, and we are charging the spores and see how the external can affect. And also, electrodes can accumulate potassium. If it's negative, then will be attracted. So we are trying to see if spores that are closer to the electrode will be then inhibited compared to the ones that are farther. But yeah, there are many experiments that we can do from this. Yeah, thank you. Yeah, exactly that. Yeah, during the formation of the spores, they can accumulate different concentrations. Yeah, we have kind of a normal distribution. Yeah. Thank you. Yeah. That's it. Okay, thank you.
Okay, as Bill said already, my name is Natalia. Hi to everyone. I wanted to thank the organizers for inviting me here today. And today I'm going to talk about some protein membrane interaction studies that we perform using spectroscopy correlation. So this is a little bit of what I'm going to talk about today, hopefully with the context of the biological subject that I'm studying and really briefly the methodology that I'm using. If I did that part correctly, you will understand the results that I want to share with you and the conclusions that we got. So just small background. I'm from Argentina. I'm doing my PhD at the University of Buenos Aires. I also got my master's from the same place. And if you don't know anything about Argentina, all you need to know is that we are the south of South America, and we're currently FIFA's world champions. So my advisor is Laura Estrada. She founded this group 10 years ago. We're all physicists. And I would say that the main goal of our group is to provide new tools and techniques to answer new biological questions. In this sense, we currently have the only homemade 3D orbital tracking microscope in the country, and we have a homemade 2-photon counting microscope, and all of our equipment are available for the community to use for free. So I'm in my PhD, I'm studying the dengue virus. This is a mosquito-transmitted disease. It's mainly located in the tropical areas of the globe. It affects about 390 million people per year. And this map is already old because for several reasons, but mainly global warming, it has spread a lot. See, this is Argentina, and it's supposed to be free of it, and that's not the case right now. And about half the world population is at risk. And the severity of this is that we currently don't have any specific treatments for this disease. Specifically in my country, we broke the historic record in 2020. Besides having COVID, we also have the highest peak for dengue. That is the gray curve in the graph. And what is most alarming is that the dashed curve is the current 2023 number of cases. So we're probably going to break the record again. So this is a sanitary crisis in my country and South America in general. So we're very concerned about this. And what I'm studying particularly is a protein from this virus. I'm a physicist. I'm not going to try to explain the whole viral replication cycle, but you can have like 5 stages from when the virus approaches the cell, it penetrates it, use the own cell mechanism to synthesize new viral particles, then assembles new viruses that will then go release and infect another cell. And the protein that I'm studying is called capsid, and it works both in the release of the genome during the infection and the recruitment of the viral RNA during the assembly of new viruses. So this makes it an excellent candidate for a new antiviral strategy. So the results that I'm going to show you today, we got them using the percorrelation function, which is a method, you can see, from the fluorescent correlation spectroscopy family, which really briefly, what we're going to do is use confocal images. Using a fluorophore, you can use proteins that autofluoresce or proteins that are tagged with a given fluorophore, which is our case. And collecting images, or you can just collect a point and seeing how are the fluctuations in the intensity collected, you can measure how is the diffusion of these proteins. You can do that with a single point, or you can do that with 2 or more points, where you can measure the time it takes a protein to go from the first one to another one. And that is the percorrelation function, very simplified, which is what I applied today. So you can scan a line in your cell and correlate the intensity collected in 2 points of that line. So we will have pixels that we collected the information over time, or these are the same pixels over and over again. And what we're gonna do is correlate the intensity. This way we will only see if a positive fluctuation at one point will be seen later on in the another point, which will mean that a protein has gone from the first to the second one.
The protein on the membrane or inside?
It's in the cell. It's in the cell. It's diffusing. It's going through the membrane. In our case, you can do this line anywhere. You can do it just in the cytoplasm, just in the nucleus. In this case, for this study that we performed, we did it crossing. So some pixels were in the nucleus, some were in the cytoplasm, because we wanted to measure the pass through. But it's useful for measuring diffusion anywhere. You can do it anywhere. Thank you for the question. So, well, the easiest case will be to correlate the intensity that you get in one point with itself, which will be this delta R being 0. And of course, the highest correlation, it's at time 0, right? If you have something at this point at a given time, the highest probability of finding it there is at the same point, that same point in time. But if now you can see how long it will take a molecule to travel, let's say 12 pixels, the highest probability of finding it there will be at a later time, if the protein has the ability to move in that direction. If those 12 pixels are, for example, outside of your cell, you don't expect to see a positive correlation. But in this case, we did, and you can see that as the, I don't know if you can see the mouse, okay, as the red zone in this chemogram, or as the peak in the graph below. So that was the introduction for the actual technique that I used for the results that I'm going to talk to you about, which is the 2D form of this technique. Let's say you acquire now, instead of a line, a whole picture, an image with a raster scan laser. And if you take one pixel, let's say the blue one that I have here, and you want to see, okay, how is the diffusion 2 pixels distance from here? So that will be the cyan one. Well, this is exactly the same for the scan line. You're just comparing the fluctuations between 2 points. But if you take an image, now there are a lot of pixels that are at 2 distance from the center one. So all of this will give you how long it will take in average a molecule to travel from here to 2 pixels distance. But now you can study how if it's different, the direction of motion in, let me recap. So you can see whether in every direction it's as likely to go the protein than another one. And with that I'm going to explain next, I think it will be a little bit more clear. So let's assume I have 2 different particles, the blue one and the red ones. The blue one can diffuse freely in its environment. So if we look at this graph from the top, it will look like a circle, because it's as likely for the protein to go in either direction. So it's a full circle, but if now we have an obstacle for the diffusion, let's say this rock, but it could be a membrane, the protein won't be as likely to travel in one direction than another. So this circle will be deformated into an ellipsis. And we will say that the blue arrow that I draw will be the most probable direction of motion for the protein. And we're going to characterize this with the anisotropy parameter, which is the difference between the long axis and the short axis of this ellipse. So if I have a higher anisotropy value, this means that there's a very predominant direction of motion for that protein. And if the anisotropy is low, it means that it has an isotropic dynamic. And what is most powerful about this technique is that even if you can see through the microscope the barrier, which in the case of the membrane, you can see it, but even if you can't, you can detect it. So this is a simulation that I made, where I have in the first column just a box with molecules diffusing freely. And in the second column, they are the same molecules, but in this case, I have put a wall in the middle. Well, it's not exactly the middle, but there's a wall there. and molecules from the gray area can't go into the black one, and vice versa. So if we look at the intensity collected, which will be analogous to looking through the microscope, you can see any difference between these two. But if we look at the anisotropy map, which is a graph where I have drawn a line in each pixel corresponding to the longer axis of the ellipsis. So in the red protein that I had drawn there, the line will be vertical, right? So in the case where I have isotropic dynamic, all the pixels have random directions of motions. You don't see any collective behavior. But if I have a wall in some parts of the cell where the simulation, the proteins can go through, around that wall, there will be a highly orientated direction of motions, and all the proteins, what would they do is avoid the wall. So you can see here that they're all going parallel to the wall. So going into the actual results, what we wanted to do with this technique was measure how was the anisotropy, how was the diffusion of this protein around the nuclear membrane. So what we did is we performed this technique. We acquired new confocal images. And in this case, what I'm showing you are the results for capsid dengue and also for capsid Zika. Zika is another virus, also with high incidency in South America. And it has a very similar structure. It actually also has the same protein capsid. So we wanted to compare, which is part of our project, if we can find any differences in this protein that is supposed to be the same, but from two different viruses. So I draw with the dashed line where the nuclear membrane is for both cases. You see that this protein highly concentrates in the nucleus, especially in the nucleoli. And what we see is that around the membrane, let's see if I can use a pointer. Ahí está. So around the membrane, the nuclear membrane, there is a high order of anisotropy, as was expected, but also what we found was that there is a gap in the anisotropy around the membrane. See this black gap right where I draw the dashed line? Well, we didn't really know what that meant. And we also saw that it wasn't the same for both of them. So why, if the protein is the same, capsid, for dengue, we have a wide gap in the anisotropy map, whereas for Zika, we have a small gap. So we didn't know what that meant. Yes.
Are you plotting the strength of the anisotropy, or are you actually plotting them from the other direction?
I'm plotting the direct. I'm plotting this line, this blue line. That will be for each pixel. I calculate the anisotropy. It will have a preferential direction of motion, and what I'm drawing in this case is what is for each pixel the most likely direction the proteins will go. That will be something like that.
But the plot is black and white. So how am I getting?
It's just direction. It's not, it doesn't have the, I don't know the word in English. It's not telling you if it's going that way or this way, just that it's this direction. It could go like this or like this. You can plot it with colors, but I thought it would be too confusing for this. No. So I didn't say because I didn't want to put too much into it, but when you plot this, this black zone is just that I'm correlating at pixels distance, in this case, 4 pixels. So this, the first pixel, let's say, of the picture, you can correlate. But the black zones here are due to the fact that when you plot this, you establish a threshold of anisotropy that you're gonna plot. So you say, if all my anisotropy, they usually use the average value. In this case, I wanted to plot some, so I lower it. But you decide, okay, all anisotropy below this threshold, I will consider it isotropic, so I won't draw it. So those are the black zones. They are pixels where the anisotropy was really, really low. Yes, so going back to this, that will mean that in this graph, I have high anisotropy around the membrane. That will be this. And inside the membrane, it's highly isotropic.
The orientation of the anisotropy from the direction in which the lines are going? Or?
Yes. So,
Little white line thing.
yes, this will mean these lines like this. Oh, I got close to the mic. Will mean that the proteins are moving like this. Yes. Any more questions? I think I will stop, or should I keep going? No, no. I mean, we're looking at the anisotropy. We got it from looking at the correlation in fluctuations. So as long as you have I mean, the laser is moving quite fast. So I don't think you have any protein that will move that fast for you to not see it. And it's so small that even if the molecule is really slow, you won't see the fluctuation in that case. Because since what you're looking for is if it's, I mean, you have a basal point of intensity. If a molecule is fixed there, it won't add up to this analysis. So if it's something that is really, really slow, it won't add up. But in the case of these proteins, the time that we acquire is enough to see.
So like in the membrane, do you think that is true isotropy rather than just very slow?
No, it's very slow, but we're comparing it to the other. So it's, if you have another thing that is much slower, yes, that it's all relative to what you consider to be slow or not. It's definitely slower than around it. That would be the result. No, not really, because we're scanning really fast. So, I mean, we personally don't use pictures for this, but there's people who use just pictures. We prefer raster scanning because it gives us a little bit more margin to change the parameters. But no, I don't think it does. Okay, yes.
Do you know how many proteins are there in 1 pixel?
In 1 pixel, I don't know the number, but you can measure it, yes.
So the idea was to actually detecting the motion of group of proteins?
Yes, it's an average. Here we're looking at the movement of the population, not single. This is all the fluorescence correlation spectroscopy method. Don't analyze single molecule tracking. That is the huge difference. You can do single molecule tracking, or you can do correlation, which gives you the analysis of the population movement. In fact, this plot at the 0 distance, so if you correlate 1 pixel with itself, the amplitude is inverse to the number of particles that you have, so you can measure it. I just not did it. I didn't do it for this work, so I don't have the number. Yes. Go.
On the left here, apart from the membrane, there's another highly isotropic feature.
Yes. No, they are the nucleoli are highly anisotropic. I mean, I didn't say it, but this protein concentrates in the nucleus and particularly in the nucleoli. So what we saw first before we did this experiment was that where it was concentrating, and where it's going, it's as we get there, it's getting higher and higher than isotropy. Yes. I was. Oh, these are a few pixels width. So we can detect it, yes. The membrane, it's about 2 or 3 pixels in our microscope width. So we can differentiate it. Okay, I'm gonna keep going, otherwise someone stop me. So what I usually do when I have no idea what's going on in the cells is go into simulations to try to understand it. So what I try to do here is perform a simulation where I have mainly two zones. This will be the black and the red one. I only put two black sides, because when in my cells, I had like nucleus, cytoplasm, and the membrane in the middle. So for me, it was easier, but it's the same. And the parameters that I'm gonna control are the concentration of protein, how fast are these molecules diffusing, and what is the probability of them entering and exiting the red zone, which will simulate being our membrane. So what I want to do is show you if any of these parameters can replicate the differences that I found for dengue and Zika. Are we gonna see or not a gap in the anisotropy around the red zone? And does any of these parameters affect the size of this gap? So first, we study how was affected by this the probability of entering and exiting the zone. So for probability 0, which means that the particles can't go into the red zone, you see high anisotropy, but the gap is closed, which is what was expected for this technique, what I show you with the first simulation that I did. If you have a wall, you can detect it, but it's just a high anisotropy line around it. But if you have a higher probability than 0 to go inside this zone, which is 2 pixels width wide, you have a gap in the anisotropy map, yes, which is what I found for capturing dengue and Zika. And this is reassuring because we know that the protein is going to the nucleus, which is what will happen in this case. If the probability is more than 0, the proteins go from the black to the red and then to the other black zone with a different probability. But the size of the gap that we have in the anisotropy map is the same for every probability. So this is not showing us the difference between dengue and Zika. So I went on and checked if maybe it was the concentration of protein. Maybe it was that in dengue we had more capsid than for Zika. And this was in it either the gap size is the same. Then I checked maybe it was diffusing faster one than the other. And again, don't get so distracted by how many white lines you have around this zone. What we're looking at is how width is the size of the gap, and they're the same for all of them. And of course, I left last the one that I like, which is that we found actually a parameter that changed the gap of the anisotropy map, which was the ratio between the diffusion coefficient inside the red zone and the one outside of it. So what I'm saying is that in this case, the molecules that are inside the red zone are diffusing 1,000 times slower than outside of it. And in the last case, the molecules outside and inside are diffusing at the same speed. So we performed several simulations to check that this wasn't just a fluctuation. And then I performed some experimental validation where I checked for GFP and a membrane-bound form of GFP, which is a protein that will go to the membrane. You can see it in the intensity map. And if you look at anisotropy for GFP, you have high anisotropy around the membrane here, here, and here, which is to be expected. This is a wall to diffusion, and you should see it with this technique. But for the membrane-bound form, you have a gap also around the membrane, here and here. So what we found with this is that if you see a gap, it's because you have a membrane that the protein is interacting with, and the bigger the gap, the stronger the interaction. More interaction means bigger gap. So going back to my dengue and Zika study, this will mean, because I saw a bigger gap for dengue than for Zika, that the capsid from dengue has a stronger interaction with the nuclear membrane than the capsid from Zika, right? So to summarize, we found a new method to measure the strength of the protein membrane interaction. And in the case of dengue, which is what I'm studying, capsid has shown a stronger interaction with the nuclear membrane than the same protein for Zika. I think I have like a minute just to talk to you about this paper that we published now 2 years ago. Here we use the 1D form of this technique, the pair correlation function, using line scan. And we did that to measure, like I said before, how was the diffusion in the cytoplasm, how was the diffusion in the nucleus, and how was the transport inside the nucleus. So we know this protein has to go to the nucleus to fulfill its functions. So we actually found that there are more than one population in terms of diffusion, the dynamics that it has, both in cytoplasm and nucleus. Some proteins diffuse freely, others are interacting with things, making it slower. We found how fast the proteins are being transported inside the nucleus. And we also found for the first time that this protein actually shuttles outside of the nucleus as well, which hadn't been shown yet. And we still have a lot of questions about this protein and its dynamic, like what is aggregation state of this protein in the nucleoli, because it accumulates there, so it's likely that it will aggregate. What is the function of this protein shuttling outside of the nucleus, which is the result that we got from the paper that I showed you just now, because there's no known function of this protein outside of the nucleus, so why would it go outside? And from what I talked to you about today, we want to check if we can find any difference in, it could be the infected cycle of dengue and Zika, or the capsid protein itself that will make it interact differently with the nuclear membrane. Because so far there is no study that shows why could this be happening. So I want to thank our collaborators, Enrico Beraton, who developed the technique that I used, and our virologist, Andrea Gamarnik and Guadalupe Costa Navarro from the Instituto del Elba. Thank you. Any questions? Yes.
Maybe you mentioned already, how do they cross the membrane?
What?
Is it known the mechanism to enter?
Well, actually, we know that it can diffuse, but the times that we got from this work, from how fast the times were, it agrees with active transport. So it could be a protein like, yeah, it has a just diffusing portion, but mainly it's active transport. Yes.
Do you know if it could be, I mean, I read that it's not possible to know if it's exactly the same for them, like, because maybe one transporter is more specific and it can do it faster, and then because of that you have the difference between, like, you have the gap.
You mean for dengue and Zika?
Yeah.
Oh, we didn't perform this study particularly for Zika, but it will be interesting to see if the times, that's really interesting what you're saying. Like if the times are different, maybe that's why they, yeah, that's really interesting. I'll check that out. Yes.
Any questions? Can you tell which side of the nuclear membrane is interacting with?
Oh.
Inside the nucleus.
Not really, because what I'm seeing is I don't think so. Maybe, but I didn't try that. I mean, we know that it accumulates first in the, how do you say it? The retoplasmatic. I mean, if I have to guess, I would say from the cytoplasm side, because we know it interacts highly with that zone. But I don't know if just by the results that I got, you can say that. I think you need another study. Yes.
Oh, so just to make sure, when you refer to interactions, you're referring to interactions the protein makes with the nuclear membrane.
Yes.
Oh.
You want me to talk more about that? Or what was the question?
Oh, I guess the follow-up question, if I look at like the long, like T time scale, you had like proteins that were like outside of the like on the other side of the isotropic region. Would you expect all of those proteins to like migrate to the membrane?
You mean, let me check. In this graph, for example?
Yeah.
Okay. So you're saying if all the proteins that are, for example, here will go here?
Oh, the other way around.
Here to here? Well, the concentration in absolute terms, yes, because this protein concentrates in the nucleus. So most of the protein will go from the cytoplasm to the nucleus, but it doesn't disappear from the cytoplasm. It's still a portion of it remains there, at least at the time. This is like 6 hours post-infection, maybe a little later, and as time go by, there will be less concentration there. So yes, it will migrate, but not necessarily to stay in the close to the membrane. It will migrate and actually go to the nucleoli. So I wouldn't expect it to concentrate around the nuclear membrane as much as going past it, interacting with it, to go to the nucleoli where it fulfills its function. Okay, thank you. Yes. You mean if there's a part of the nuclear membrane that is interacting more than another? Yeah. I would expect it, yes, but I haven't found that. I mean, I think that the variability that I get, because I don't find I didn't plot that, but I'm gonna use this as an example, but it's not from the data, but I got something similar. Here I'm plotting what is the size of the gap for different simulations and different portions of the simulation. Let's say I measure it here, here, here. I did the same for dengue and Zika for my actual experiments. And there is a variability, even in the simulations. And I think that the variability that I can see will be most likely to fluctuations. Not so much, I don't know if you can measure it. You can measure it if it's like really large, the difference. Let's say if the difference is between one of these graphs and the other violin plot, maybe you can see it, but if it's small, I don't think you can. I'm gonna, okay, that's it. Okay, thank you.
Okay, so I will talk about stochastic thermodynamics. I don't know how familiar you are with this concept. So it's basically to describe really small systems where fluctuations are super relevant. So you cannot just use regular thermodynamics because they don't account for any fluctuations whatsoever. And I also study non-equilibrium systems. And the main thing of my talk and my work in general is that when you want to apply stochastic thermodynamics to real life systems, usually you have to make lots of assumptions, and then you ended up with a setting that you cannot apply stochastic thermodynamics anywhere. And one of the things is that you need to monitor all the relevant degrees of freedom. So let's say you have a process. I will use the jump process, which is a continuous time process, a continuous time Markov chain in discrete state space. So this is a graph, and every node of the graph is a state, and every arrow is a transition between two states. So this is just some level of coarse graining where you take the mesoscopic states, the relevant mesoscopic states, but often you cannot just observe all of these states and all of these transitions, and you have to deal with the partial information that you collect from your experiment. And the status quo of approaching this problem is that there are some techniques like lumping, decimation, milestoning. They are coarse graining methods to deal with this hidden information, but they all focus on the states. So for example, lumping would be I mix this state with this state, and I cannot distinguish between them. But in many systems, like when you have a molecular motor, and the molecular motor is stepping forward or backward, what you are observing is a transition. So a transition between two states regarding the position of this molecular motor, but you cannot observe all of the hidden transitions that are pumped by chemical transformations. So you are not observing any states. So let's say, suppose this describes a molecular motor and this is one step forward. After a step forward, maybe a chemical was absorbed and then the system is now here, but you don't know when the system left this state, so you are not observing states whatsoever. The same goes for the ribosome or like one electronic device where electrons are being pumped from one side to the other, and then you observe electrons hopping, but you don't observe what they are doing in your device. Okay, and this is really important because I want to do some model-free inferences, which is, I think, a big step that we have to take in stochastic thermodynamics, where you're not making some inferences that are really conditional on the model that you choose. So you can make some thermodynamic inferences without assuming a model. Okay, so in the beginning, I would just make a baby stochastic thermodynamics speech for you, and then I will talk about transitions and only the visibility of transitions. And then I go back to stochastic thermodynamics and some implications also to biophysical systems. Okay, so stochastic thermodynamics. There are two main things in stochastic thermodynamics: the fluctuation relations, which is particular to stochastic thermodynamics, and the entropy production, which is the main quantity of stochastic thermodynamics, and it's also a measure of dissipation, of irreversibility. It's a measure of the distance to equilibrium, and the two of them work together because the relevance of each other is tied together, and with them we can obtain the generalization of the second law to non-equilibrium and small-scale systems, the thermodynamic uncertainty relation, which is a milestone of stochastic thermodynamics, the Jarzynski equality. We can define what is thermodynamic consistency. So if you define a model, you want it to be thermodynamically consistent, and also some thermodynamic limits for what you can do and what you cannot do, just based on thermodynamics laws. Okay, so the entropy production is many things, but it's also the thermodynamic cost. So for example, suppose that you have a system and you want to make a transformation between two equilibrium states. So like these are DNA strands where you are pulling and pushing them. And the work required to go from one state to the other state, if you go from in one equilibrium pathway, it will be the free energy difference. But if you do it in a non-equilibrium way, so if you do it too fast, for example, then you have to sum the temperature times the integrated entropy production. This is the integrated entropy production over your experiment. Another example is a class of molecular motors where you have like a molecular motor doing some work against a load force, and the rate of work, the chemical work, or say the power that you need to spend through these chemical transformations to keep your motor with a velocity v against this load force F, it is given by the power is F times V plus the temperature, and here's the entropy production rate. So the difference between this big sigma and the small sigma is that this is the integrated version of this. And then we define the efficiency of this molecular motor as 1 over 1 plus temperature, entropy production, and FV. So it's really important here because here the entropy production is appearing in the efficiency. So it's setting the thermodynamic cost for the operation of this molecular motor. So if you have like a lot of dissipation, then your efficiency is going down. And if you have no dissipation, then the efficiency is Carnot. Okay, so yeah, I just wanted to highlight one of the important aspects of stochastic thermodynamics to biophysical systems. But what exactly is going on in these systems? Maybe you are already familiar with all of this. Basically, the jump process is defined as a vector of the probabilities of each state. And the time derivative of these probabilities, say the population of each state, is given by this transition rate matrix. So the entries of the matrix have the transition rates for all of these black arrows times the probability distribution over the states. And the solution, the formal solution of this equation is that the vector with the populations, time dependent, is this thing we call the propagator, which is the exponential, the matrix exponential of time, times the transition rate matrix, times the initial distribution over states. And then we define a set of visible transitions. I call it calligraphic L. And the visible transitions can be, like in this example, the transition in this direction and in this direction. It can be anything else. I'm not making any assumptions regarding which transitions I'm observing, just a few of them. And I use the notation that this small L is a transition in L, in calligraphic L. And then this transition is, of course, pointing from one state to the other state. So when I use double bra and double cat, I'm saying this is the origin of this transition, and this is the tip of the transition. So if the transition is going from here to here, double bra L is this state, and double cat L is this state. Okay, so if you, yes? Yes. Yes.
But as you said, you don't know the chemical state.
Yes.
So in particular, when it's step from n to n plus 1,
yeah,
you don't know that transition could be associated with many different chemical states. So
yes. So it depends on what you are assuming regarding this molecular model. So let's say the kinetic model, where you have like 2 cycles and a step in between of them, so a transition in the middle of these 2 cycles. So there's only one transition leading from one state to the other, and this transition that is the transition that is performed when your molecular motor is performing a step. So you are observing the transition, but not the state. And there's only 2 states regarding this transition, between this transition. Yes. Yeah. Yeah, exactly. Yeah, there can be many transitions that are mixed together with whatever you are observing. It's a set of single arrows. Okay, so if you are the experimenter and you only observe transitions, the trajectory you collect is basically comprised of these intertransition times between transitions that you observe. So here you start your experiment, you wait for time t0, and then you observe the first transition L0. And then you wait for time t1, and then you observe L1. And then you collect, this is everything you can collect if we assume that you can only observe a few visible transitions. So in this example, let's say that this black line is your trajectory in the state space 1, 2, 3. And then I assume that I can only observe the jumps from 1 to 2 and from 2 to 1. And the black line, the observer cannot monitor. So this jump cannot be seen, and this residence time cannot be seen. But this jump here from 1 to 2 is what is giving a signal to the experimenter. So the experimenter basically collects. I wait t0, and then I see plus here. And then I wait t1, and then I see plus again, t1 and plus. And then I wait t2, and then I see minus. And one catch here is that if you want to see the time reversal of this, so you have to perform this trajectory in a reverse manner. Then where you have previously seen minus, now you see plus. So whenever you want to do some time reversal of your trajectories, you have also to consider the reverse of the transition. Okay, and how to deal with this hidden information, how to tame the hidden information, and the answer is the first passage time theory. So first passage time theory is just a way of measuring when is for the first time I will arrive at one state, at one given state. So let's say I want to observe transitions from 1 to 4 and 4 to 3 in this system, and I cannot measure the first transition time. But if I include 2 auxiliary states that are absorbing, and I just need to measure when is the first time that I will get to one of these states, and then I will be stuck here forever. If I measure the first passage time to reach S1, which is the first sink, it's equivalent to the first time I perform this transition. So I just need to solve the first passage time in this auxiliary dynamics. Yes? Yes, so I'm not measuring the first passage time of getting to state 3. It's just of performing this transition. And here is the difference is important, because if I measure the first time that I go to 3, it can be from here or from here. But I want to observe this transition.
You're going to estimate that with what?
With the first passage time to reach S2.
Because it's
From whatever. But yeah, the first time that I reach S2, the only possible way is by performing this transition. So I keep the same transition rates here and here. Okay. Okay. Yeah, so the structure of the solution is basically I have this survival matrix. And by survival, I mean that this is the matrix that accounts for the transitions that are hidden. So the transitions that the system can perform before doing any of these 2. So I basically take my rate matrix and I remove the transitions that are observed. So here from 1 to 4 I observe, and this is where the transition rate of 1 to 4 should be, and now it's 0, and the same for 4 to 3. This is a survival matrix, and basically the solution of the probability that I observe, given that the previous transition that I observed was Li, this is the probability that I observe the next one Li plus 1 after time t, after some time between t and t plus dt, of course. This is a probability density. And this will give me all the statistics that I need, because all I need is the probabilities of the next transition, the sequence of transitions, and these intertransition times, because this is everything I can collect from my experiment. And this is given by this weird expression, but maybe I can build some intuition by saying, okay, the last transition that was performed was Li, so I start from the tip of Li, and then I have, again, this idea of a propagator, where before was the rate matrix here, and now it's the survival matrix. So I'm propagating from this state to this state without performing visible transitions. And then I multiply by this, which is an ugly way of writing the transition rate of Li plus 1, which means that I'm actually performing Li plus 1 now. Okay, and with this solution, which is the exact solution for the probabilities of sequences and of transitions and the intertransition times, of course, I can marginalize away the time, and then I have the probability of a sequence of transitions. And then I can do the opposite and stay only with time, and then I have the probability of these intertransition time between 2 given transitions, and now everything I have to use is the survival matrix. Okay, I need 2 extra ingredients more to get to my results. One of them is the visible traffic rate, which is the expected number of transitions per unit of time in your experiment. I can also get it from the theory. And the stationary probability of a transition. So it's saying, given, you just observe the transition. What is the probability that this transition is L? This is what it's saying. And with this, I can come back to entropy production and do some entropy production inference. One definition of entropy production would be the limit of really long trajectories, 1 over tau, this really large tau, and I sum over all possible trajectories in my system. I weight the sum by the probability of the trajectory, and then I compute here the log ratio between the forward trajectory and the backwards trajectory, which would be the time reversal of each other. This is the one definition for entropy production. But I cannot compute these trajectories because they have all the information, all of the information that from the hidden part of my system. But what I can compute is the visible entropy production rate, which has these coarse-grained trajectories that are comprised of sequences of transitions and intertransition times. And this is a lower bound for the real entropy production rate. And then you can use it to infer some things about the real entropy production rate because it is a lower bound. It is really important that it's a lower bound. Otherwise, I wouldn't be able to say anything about the real entropy production. And we can split into two contributions. One contribution from this sequence of transitions and one from the intertransition times. Both are always non-negative, so either one of them can give you some information about the real entropy production rate. And also the thermodynamic uncertainty relation is a really famous result that is used to lower bound the entropy production rate. So you can have here instead of the visible entropy production rate that I just defined, you can have the thermodynamic uncertainty relation. But a conjecture that we have is that this guy sigma L is always larger than the sigma that you obtain from the TUR. So this is an improvement from this famous result, the thermodynamic uncertainty relation. Yeah. This is always larger than the sigma that you infer from the TUR. And sigma T has no relation with it. So the TUR is basically relating the integrated current, the average, and the variance of this current. And you use this information to infer the entropy production rate, right? Because on one side you have these values. And here what we are using is the probabilities of each and every transition contributing to this current. So in a sense, the TUR only considers the integral with the sum of all of these pieces. I have more information here. Of course, if I sum all of these pieces, then I recover the TUR. I just say it's a conjecture because it's not proven, but people are already using this idea. Okay, and here are the expressions, of course, for the result that I just showed. So the sigma L is given by this guy here, which has the probability of transition L after L prime, probability of L prime itself, the log ratio between these two. And for the contribution from intertransition times, I have the Kobeck-Leibler divergence between these two intertransition times. Okay, so now I will start showing this, how it performs. So for instance, if you have this system with 1, 2, 3, and 4 states, and you observe from 1 and 2, the real entropy production is the black line, and the inferred entropy production using this method, which is indeed a lower bound, is here in blue. And the magenta line is the TUR, and also the TUR has some problems because if you stall your current, the current you are observing, you can be either in equilibrium, so that's why the current over here is 0, but you can also be in a non-equilibrium system where it's very special because this current is 0, but there are other currents flowing through your system. And with this technique that I just mentioned, you can infer some dissipation. Okay, and this lower bound can also be saturated, of course, and then we understand when it's going to be saturated. It's when you have one visible edge per cycle. If you divide your graph into cycles and you put one visible edge for each one of them, then you have the saturation of your lower bound, and you measure the real entropy production rate just using those expressions. There is some evidence, so when this quantity is time independent, it's one evidence, not sufficient, but it's evidence that you are in such a regime. And in this case, the visible entropy production rate will be a sum of the sequence contribution, the time contribution will vanish, and this is equals to the real entropy production rate. And measuring the entropy production rate is a big thing in systems because it's not trivial how to do it. You cannot just put a device that gives you the entropy production rate. And in this case here, for a ring network where you have one cycle and one edge in this cycle, visible edge in this cycle, here the entropy production rate in black, and the dots, the blue dots, are what you are inferring. and they are precisely the same, and the TUR is not inferring all of the production rate. I think I have to go a bit faster. So going back to that example of the class of molecular motors, where the efficiency is given by 1/1 plus T at production over FV. If I have one lower bound for the entropy production, then I can have an upper bound for the efficiency. So if you observe these transitions, these steps of the molecular motor, you can build one upper bound for the efficiency of such a molecular motor. And this has been done using the TUR, but now you can do the same using something that is tighter than the TUR. And here indeed, you see the real efficiency of your molecular motor for different load forces. And here the upper bound by the TUR, and here the improved bound using the expressions that I just showed. And also we have information about the many details about the statistics of these transitions and the intertransition times. And it can also be used for instance, let's say you have a model with disorder. So there is a molecular motor translocating through a strand, and it has monomers, and these monomers are random. Every time you are in a different monomer, the transition rates change. And then we realize that the intertransition time, the asymmetry in the intertransition time between two forward steps and two backward steps, the black line is always 0. They have the same distribution, but between plus minus between a step forward and after a step backwards, and minus plus, it changes. So in a way, you are probing the disorder. If you have a way of changing the disorder parameter, maybe by chemostating something that is affecting the disorder, you can probe the presence of disorder just by looking at these statistics. And this I want to skip. And in summary, we developed a framework for coarse graining that is based on the occurrence of a few visible transitions. And with this, we can do some model-free inference. So we were inferring the entropy production rate without making any assumptions regarding the model that is underlying the dynamics you observe. And we can do also the opposite. We can do inference of the model. So we collect the data and we see from which model I expect to observe this data. And you can do some inference of the model. So for example, I just said that if you have one edge per cycle, then you're gonna, that quantity that I just showed will be time independent. But then if you measure and it's time dependent, you say, okay, so there are some more hidden cycles that I'm not observing. And you can do inferences about the dynamics, the thermodynamics, biophysics through the disorder or many other things, and the topology and the structure and the underlying structure. And I didn't say much, but we can also use this framework to recover a fluctuation relation when you observe just a few transitions and not all of them. And with this, I finish my talk. Thank you. Yes.
Let's say I have like some experimental data, and is it claim then that if I were to use your formula, I would always get a better lower bound?
Yes.
Than if I were to use the normal uncertainty?
Yes. Under the assumption that your data is coming from transitions, okay, and you can distinguish these two transitions.
And what would be data that tells?
I don't know if you can observe in one state.
Oh,
yeah. Yeah, if you have like one electron in one position, and then you can see when the electron jumps out of it. Yeah, you are observing the state. Yes.
But in your example, it's like, well, I could make this step, but I don't know what the rest of the molecule is doing. So it's not really those often in biology, I guess, those transitions.
Yeah.
Wow.
Yes.
Deal with that. I mean, it's bound, or I don't know. That more general.
About the more general situation, we'll need to do some relaxation of the assumptions that we have in the system. but maybe it clarifies a bit if I show the kind of model that we have in mind, and that there are some papers by Carlos Bustamante where they show this class of systems where you have like a set of states, and then when you perform this jump, it means that one ATP was absorbed by your motor, and then when you perform this jump between two specific states, it means that a step forward is taken, or between from 5 to 2, a step backwards is taken. So this is the kind of class of models that I have in mind. But then if you have more transitions and you cannot distinguish between them, there are many ways you can characterize them. Let's say you can have this transition, but then you have another transition where the chemical has a different concentration. Then you are mixing these two in a single transition that you observe, which is the step forward and step backwards. And this is the ongoing research that we are doing, how to deal with this kind of systems. Yes?
Yeah, I don't know if this is related to that question, but like, what if states are no longer discrete? And so
Okay.
like continue
Yeah.
all this. Is there some sort of generalization or something that you can say about that?
Yeah, in this case, it's a bit more tricky to define what is a transition, and because you have a continuous state space. And for this case, I wouldn't know how to define the transitions and how to run the same analysis. So I say that I just don't see how it would apply. Yes?
So you mentioned that if the number of transitions that are observable and the number of loops are equal, pressure is down?
Yes, but the transitions you are observing have to be sitting on each one of the cycles.
Yeah, you know how many transitions they are independent.
Yes.
Then it is exactly for the rank.
Yes. Yep. Yeah. No. In the class of models that I have in mind, the states they are hidden. They are naturally hidden. So this is precisely what I'm talking about here. Thank you.
Thank you very much. I have to say hello, everybody. Thank you for having me. I wonder if this actually works. Oh, wow. Okay. So thank you once again. I'm Kabir. I'm a postdoc in Arvind Murugan's group at the University of Chicago. And today I'll be telling you about some ongoing work, actually, we're doing on, well, what we think could be described as the evolution of kinetic proofreading. And it's work that's going to begin with a proposal, shall we say, from theory. And then I'm going to tell you a little bit about the experiments that we're doing to test that idea. Okay. But I first want to begin with a broad introduction to the class of questions we're engaged with here. And that is, who let the demon out? We're familiar with the idea that in the physical world, you can't maintain order very long. If I collect all the molecules in one side of the room, I come back a few minutes later, they'll be diffused throughout. Right? The second law is quite powerful. But we noticed for a long time that living matter, broadly speaking, seems to somehow get around this. It starts in order configuration. I come back a while later, a year, 2 years, 5 years, and that order is maintained. And here, what I mean by that is it's quite simply remarkable that my ribosomal RNA gene looks recognizably like E. coli's ribosomal RNA gene, despite the fact that they're millions of years apart. And of course, there's sort of a broad understanding of how this is possible. It's because there are mechanisms in biology that dissipate energy to fight off the second law and maintain that order, right? And these are often studied today. Their function is studied often by the kinds of techniques and ideas in the last talk. But the question I want to pose to you today is slightly different, which is, how did these mechanisms come to be in the first place? What drove them to emerge? And any sort of question of this type, like why do you see X? There are, broadly speaking, two classes of answers. One is sort of the standard Darwinian answer, that you see it because what it does, in this case, promoting order, is useful. And so selection acts on it, and you fix it. It's because it has a fitness benefit.
The other class of answer is that no, it's got nothing to do with evolution or fitness benefits. It's because physical constraints demand it has to be so. And in the case of demons, there's some ideas by Jeremy England, maybe going back to Prigogine along these lines. But what I'd like to suggest to you is something that's slightly different today, and that's the subject of today's talk, which is that demons of the type I've described emerge through evolution, but not because that order has any benefit. They can arise even when that's not the case. Okay? And in the strongest version of my argument, I'm going to say that it's an inevitable consequence of self-replication. It just has to happen. And the particular demon I'm going to be interested in today are DNA polymerases, which are the machines that are in great part responsible for the fact that genetic material is passed down from us to our children faithfully. Makes a mistake, meaning putting a C across a T instead of an A once every billion bases or so, depending on the system. And of course, the way it does this, sort of simply put, is because it implements a mechanism called kinetic proofreading, which is sort of a well-studied, very canonical demon that consumes energy and at the expense of time reduces errors. It's a multi-step process. Okay, but before I dive into this, I just want to say that the work I'm presenting is done with lots of folks. Arvind, of course, but it's in close collaboration with a brilliant postdoc, Ricardo, who came in as a theorist and who I've press ganged into the lab recently. Samana, Naija, Vedant, and Finn have contributed a lot to various aspects, and we've benefited a lot from conceptual and technical assistance from particularly our polymerase expert Marco in Paris, but also Jack, David, and Rama at U Chicago. Okay. Let me start with theory. So if you write down any model of kinetic proofreading that you might care to, and there are several out there, and you analyze it by the methods we saw in the last talk, you will typically find that there's a trade-off between the time it takes to undergo a certain, your catalytic action and the fidelity of that action. If you want to be high fidelity, meaning at a low mutation rate, if you're thinking of a polymerase, you need to spend a long time to do your actions. And this is sort of intuitive in the sense that to do something well takes a large amount of time. So we went into this and we actually added one small thing that was biophysically motivated. It seemed very innocuous. We didn't know it had the effect it did. Was to consider the fact that mistakes, especially when you're thinking of template copying, are not just changes in letters. You've put the wrong molecule into a growing strand of DNA. It doesn't sit well against the template strand. As a consequence, it's not sitting quite right, and there's a structural mismatch. So the next base has a hard time inserting itself in. Actually, even worse when you think about the fact that this is inside a polymerase, which has a pocket that's shaped to accept matched base pairs. This gums up the polymerase. Okay, and so this slows down replication, usually in measured numbers, about 100-fold. This has been seen in a variety of contexts, both enzymatic copying as well as where we first heard about it, non-enzymatic replication. So it's just a shape mismatch. And so you might appreciate here that if you had mechanisms to correct or to prevent error, even though they take more steps, depending on the balance of timescales, this can make the entire thing go faster. Right? That's a simple idea. But of course, it's a balance because this is slow, this has multiple steps. What happens when you combine them together? And so if you stick it into, again, generic model of proofreading, you have a curve like this that characterizes the proofreading model. You have a curve like this that tells you how long you stall as a function of your mutation rate. And you stick them together and you get a U-shaped curve. Just say there's a region over here where faster is more accurate. Okay? And of course, there's a region where there's a trade-off. And just to make sure that I'm communicating the essence of this right, no curves or anything, I'd like to give you an analogy. Pretend I was teaching a class. And pretend, which may or may not be the case, that I'm a particularly bad teacher. And I want to get out of the room as fast as possible, so I give the students an assignment. And the assignment is, copy down 100 words from this essay. You have to copy it down from your textbook, but I will not read it. I will give you grades depending on how fast you finish. Okay? And I want to contrast two students, a proofreading student who's very diligent. They check each word twice before writing it down, and a non-proofreading student who just copies down as fast as they can. Okay? And I tell them to go. And naturally, the the not proofreading student zips away. They're going much faster. But then disaster strikes, and the non-proofreading student sticks in the word banana, which does not appear in this text by Darwin. And that totally throws them off, because now they look back in their text and they cannot find any mention of banana. They don't know where they are. They don't know what to write down next. Meanwhile, the proofreading student diligently marches on, keeps on going. At some point, this person figures out where they are, can start moving again, but by that point, the proofreading student has already won the race. Okay. All of which is to say is that if mistakes cost you time, then being able to proofread to error correct can lead to speed, can give you speed. All of that is fine. But now I want to twist that around. If proofreading gives you speed, can you get proofreading by selecting for speed? Does speed give you proofreading? Okay. And so Ricardo did a rather imaginative simulation. He started with a random Markov network as a model for a polymerase, initialized at equilibrium, many states, many transitions. This thing doesn't proofread in any sense. And he put it through a simulated evolution where he said, well, the only thing that I am selecting for is the ability for this to copy a strand of DNA as fast as you can. Okay? The time to copy a strand, of course, goes down. But the remarkable thing is that if he puts in stalling, this idea that making a mistake makes the next base slower, he finds that the mutation rate also goes down. So faster is more accurate. The system automatically or spontaneously breaks detailed balance, starts consuming energy. And like the diligent student, it takes longer to insert each base to make sure that it doesn't make the mistake that causes it to slow down, right? So a simulated evolution experiment where you select on speed and you get proofreading fidelity as a consequence. Okay. Now this is sort of it seems, yes? Yes. Otherwise, you will not couple to them. That's precisely right. And depending on the because there could be cost to coupling to them, it could be rare. I agree. And so this is fantastic because at some point I'm going to say we should just go to experiment and test it there. And of course, these things are at play there. So we'll hope to get to that. Okay. But maybe pursuant to this, I'm going to try and generalize this a little bit. Okay. In some sense, I described it to you in terms of enzymes and polymerases and DNA, but it's maybe an even simpler idea than that. Imagine instead of copying a strand of DNA, the thing the cell needs to do before it divides is assemble a viral capsid out of many different components. And these components have off-target interactions. And because of these off-target interactions, which are weaker, they can insert the wrong protein in the wrong place. That slows down the protein that comes afterwards because it can't make the right connections. Okay? But in this system, you can imagine that if you evolve, if this was available to you, if you evolve something like a dynamic instability, that regardless of your configuration, just shunts you back to nothing built at all, this would act like a demon. It would increase the chance of getting the right structure. But more importantly, you could get it just if you ask this thing to be fast, because it would also increase the speed. And there's some way to generalize it, and we're trying to do it that way. This is my last theory slide. You could imagine instead of thinking about particularly self-assembly of proteins or copying DNA, there's just some set of tasks you have to do to go from a newborn cell to a replicated cell, let's say. There are many different paths you can take. And perhaps it's true, and this is the thing we put in, perhaps it's true that some of those paths are much slower than other paths. In the case of DNA, this is sequence-based. Yeah. So in terms of this division time distribution, then, you can write down what the exponential, long-term exponential growth rate of this population of cells is in terms of a demonic resetting, something like I described in the last slide, which is you just at some rate, regardless of where you are, come back to the beginning. Yeah. And then you can ask, as a function of parameters of this distribution, when is this resetting worthwhile? Meaning, when does it actually increase the growth rate? And you can do this, and you can find that as a function of the variance of this distribution, which is rather intuitive, at some point it's worthwhile evolving a reset daemon just to speed you up. But of course, it gives you something that looks like order because you exclude some trajectories and you keep others. Yes?
Do reset rings have to go all the way back?
No, 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 no. It's just if you want to do a calculation, it's simpler that way, but no. And I think this is, again, it goes back to Bill's question. What's the space of mechanisms you have available to you to actually do? This is one thing that you could imagine there is, but no. Doesn't have to be that way at all.
In the DNA system, is the reset only one?
Yes, yes. In the DNA system, yes. I think possibly RNA polymerases that it can move back further. I'm not sure. The DNA we think of as one back. I actually don't know if direct single molecule experiment showing that. There must be, but I don't know. Okay. Where was I? Ah, yes. So depending if this distribution is very broad, you'll evolve a daemon that resets just for speed. Okay. So this is some theory we did, we're still doing, but the proposal we thought was really worth testing was just this claim that if you take a system and you select it for speed, if you have some notion of stalling, you can get order, meaning fidelity. Okay. And is there some way we can test this? So I should say as a disclaimer, we haven't actually done the experiment yet, but we solved some challenges along the way that are quite interesting, and I'd like to tell you about them. Okay, so this is the experiment, and as the title suggests, it involves Luria and Delbruck. Okay, the experiment we would like to do is the following. It's a directed evolution experiment of DNA polymerases. Imagine you started with thousands of DNA polymerases whose mutation rates you know. You have to go out and measure them. You then select on this library just for speed. You pick the fast ones only. And you say, which of the polymerases make it through? And in particular, do I only get the high fidelity polymerases out? Simple experiment. Take polymerases, select for speed. Do you get the high fidelity ones or not? Okay. There are two experimental challenges to actually doing it. One is, how do you actually do this selection experiment on just the speed of a polymerase? And two, which I'll discuss at the end, how do you actually measure thousands of mutation rates, which is the input data I need? Okay, why is it difficult to do directed evolution on DNA polymerases? The DNA polymerases live at the core of your cell. They copy all your DNA. Any changes to them will ripple down to changes in all the thousands of genes they copy. And so it's hard to do build a well-controlled experiment. And this is why they're not routinely evolved. But what we read about just about when the pandemic was starting was this fascinating system in some yeast. It turns out some yeast have within them an odd cytosolic plasmid. And it's odd, well, aside from being cytosolic, it's odd because it's copied by a dedicated DNA polymerase. Okay? It's a small thing, only 10 kb. And the interesting thing about it, the thing that caught our attention, was that in this system, it's easy to tease apart the traits of the polymerase. In particular, it's a multi-copy plasmid, and it's been shown that the speed of the polymerase sets the copy number of the plasmid. In contrast, the fidelity of the polymerase sets how often you get mutations in genes encoded there. And this is in yeast, which of course we have a lot of tricks for. And so we can use well-established methods from the yeast world to build an experiment where we take this plasmid, we get rid of whatever junk it had on it initially, and we put two genes in. One gene that allows us to select for the speed of the polymerase through the expression level of this gene, which is related to the copy number of the plasmid, in a dose-dependent way, meaning we can add a drug and do it. And the second, we put a gene with a stop codon that the yeast need to grow in media without leucine. And so if you drop these cells in minus leucine, you only get colonies if you fixed this stop codon, which of course is related to the mutation rate of the gene. Okay? And just to give you a sense of how powerful the system is, it turns out that another group had actually built some of the data we wanted to try and answer this question. So this is data I'm showing you from Chang Liu's group, where they in fact had made about 100 to 200 DNA polymerase variants for a totally different reason, not our question, and characterized the copy number of the plasmid, which is the proxy for the speed, and the mutation rate of the DNA polymerase. It's actually a rather unique data set. But the thing that was really pleasing to us was the fact that it does seem to show this counterintuitive trade-off that I was trying to motivate at the beginning of the talk, which is the faster polymerases are more accurate, and the mutagenic ones are slower. Okay. Now, there are many caveats with this data set, which is why we still want to do our experiment. In particular, these weren't mutations chosen at random. There's no way a simulated evolution experiment, because these mutations were chosen with some structural logic. These folks had a certain goal in mind. But it's encouraging. Okay. Which is to say, this is a great system for us to do the experiment we want to do. That's the first challenge. Second challenge is a little trickier. How do you actually measure thousands of mutation rates? Well, let me just step back and say, how do you actually measure mutation rates? When I got into this, I assumed that you do it by sequencing. Turns out that's not very good, because the error rate of sequencing is much higher than the mutation rates you're trying to measure. There are tricks that get around this, but they're difficult and expensive. It turns out that the best way still to measure mutation rates, if you can do it, is the 1943 Luria and Delbruck assay. And just to briefly introduce you to what that assay looks like, if you Luria and Delbruck, 1943, they did the following experiment. Take a single bacterial cell, put it in a tube, and let it grow to saturation, millions of cells. In that process, it generates mutants. You then plate it onto an agar plate that has, say, an antibiotic, and only the antibiotic-resistant mutants survive. And you can count them, count colonies. Okay. Clearly, a more mutagenic bacterium will produce more colonies. And in particular, what you do is you have to repeat this many times, because it's a very stochastic process. You build a distribution, and you fit that distribution, which typically is assumed to have only one fitting parameter, which is the mutation rate mu. In particular, there's lots of theory, an entire lineage of theory that has spent time solving this process, and it turns out that this distribution famously is very broad and has a power law tail. This will become important in a couple of slides. Okay. So this is the famous Luria and Delbruck fluctuation assay. Some of you may have done it in class. I hope you haven't had to do it too much, because it's really laborious. What you have to do, because you have to build a distribution, is for everything you want to measure the mutation rate of, fill up, say, 50 or 90 replicate cultures, each one with one cell, let them grow to saturation, and then one by one spot them onto an agar plate and count colonies. It takes a long time to say, takes even longer to do, and that's just for one number, one mutation rate. Okay. It's basically how they did it in 1943. We decided that, well, this is the modern era, and we can do better. What you can do instead is replace growing replicates separately with DNA barcoded replicates. Each barcode acts as an independent statistical replicate of this experiment. And you can pool it all together and do it in one flask. And here we're not limited by the patience of the graduate student or postdoc doing the experiment. The thing that sets how many barcodes you can put in is the transformation efficiency into yeast. And we're really good at dealing with yeast, so that's high. It's at least 10,000, but more recent experiments, it's more like 1 million. And the point is, you do the same experiment, but instead of counting colonies, you just sequence the barcodes, and you infer how many mutant colonies there were. So we call this a high-density Luria-Delbruck experiment by sequencing, which is a mouthful that abbreviates to hide and seek. Okay, so like I said, we decided that the first thing we wanted to do, this is the last thing I'll show you, with this technique was to ask, does it actually work? Can we actually do anything interesting with it? And we decided we'd go after and see, can we actually see this power law decay? The best measurements we know of this distribution are from Andrew Murray's group about 15 years ago, where they had 700 replicates, and they still plotted it on linear axes, so you can see very much. So just to orient you, I want to show you some data. You're going to actually see two curves showing up, each one 50,000 barcodes of yeast. The reason there are two is because this was actually for a project that I'm not telling you about. There are two different variants of yeast, one with and one without a chaperone. I'm not going to tell you why we did that. But basically, you barcode them, you grow them up in the biggest flask we could find, which was down the hall in the protein biochemistry lab. And then you select them onto an antibiotic, sequence, and count colonies. And here are the distributions. And you can see two of them. In gray, I've got the expectation from the Luria-Delbruck distribution. This large regime over here is the power law that you expect from Luria-Delbruck. There's some interesting deviations out of the tail, which is actually what we were looking for in this experiment. But I won't describe that today. It has to do with the effects of chaperones on promoting standing genetic variation. But for the purpose of this talk, I just want to say that this method seems viable. I think it gives us a way to measure mutation rates in much, much higher throughput than we've been able to before. Yes, correct. 5 minutes? Thank you. Yes, you infer from that. So if I can also I think I can translate your question into you're only measuring mutations that give you resistance for whatever antibiotic you put on. Yes. The trick is to choose your antibiotics such you know how many mutations give you that resistance. And then you work out the mutation rate in the DNA. You have to choose the system to do it carefully. But basically that can be done. Oh, yes, they do. And so we don't actually sequence the mutation itself. We just sequence the barcodes. Exactly. Which is why we use a system where we know exactly where the mutations are going to be. Makes everything much easier. Okay, I'm running at the end of time. I just want to conclude with maybe just a restatement of what I've been telling you, or perhaps to put it in a different language. There's a question that we've become interested in, which is slightly orthogonal to what I talked about, which is maybe a bit funny, which is why are mutation rates sometimes really, really low? I've got some numbers on the screen. Humans are here, more interesting organisms are down there. And I've put it up there, mutation rate per base pair and the size of their genome. And what I want to try and illustrate from this is that sometimes these numbers are reasonable. Reasonable in the sense that you're getting about 1 mutation every 10 generations or something, which is about what you'd expect if you were trying to get rid of deleterious load, or 1 in 100. Something over here, on the other hand, you're getting 1 mutation every 100,000 divisions. And it's difficult to see how you could account for that if the selection pressure keeping mutation rates this low was simply the need to avoid deleterious mutations. So maybe if you've bought what I've told you so far, if we buy what we've told you so far, perhaps there's an alternative possibility, which is that actually these mutation rates are kept low, not because evolution's worried about deleterious mutations, but instead because evolution wants to copy as fast as it can. A lot of these are DNA viruses where DNA replication is the limiting step. And I guess this brings me to this idea that perhaps many of you know of, the idea of a spandrel from Gould and Lewontin. So if you don't know, this is the Cathedral of San Marco. These are spandrels. These places seem like absolutely perfectly made to park a painting of an angel. And you might imagine, wow, the architect must have really worked hard to put these in. But in actuality, they're just architectural engineering consequences of having to support this dome. You need these there because of physical reasons. And it just so happened that people realized they could be used to put paintings up. And so the idea of this paper, and I think the idea that is particularly interesting, is sometimes you see remarkable things in nature that you think, oh, it must have evolved because of all the remarkable things it does. And what I'm trying to suggest maybe is that things like the high fidelity of DNA replication could, in some regimes, be something like a spandrel. And with that, I'll thank my funding. Thank you for your attention and close. Yeah, it's so you're absolutely right in that you do need that the position of that minimum is set primarily by the time penalty. I do not think that we have gone and looked to see exactly what fold slowdown you need. I don't know. There's a challenge here, which is the difference between the bare mutation rate of a polymerase, which you have no access to, and the mutation rate you actually sequence. And it's the bare one that's relevant, but it's the sequenced one we can measure. Yeah.
For example, are you claiming that it is dependent only on?
Good. So the claim here is that whether or not resetting is advantageous is dependent only on the distribution of completion times. That is correct. Whether or not that gives you order in the sense of excluding a large amount of trajectories, certainly not.
And so, variance would be
Correct. Mean is at 1 here.
and what's the
Okay. This is great. So what the data I'm actually showing you here, it's interesting, we don't fully understand this yet. The data I'm showing you here is for a log normal distribution. I've set the mean to 1, I've got the variance. It turns out the shape of the distribution matters a whole lot. In particular, it does need to have a heavy tail. For instance, you could have a Gaussian distribution of completion times, and resetting is never advantageous.
For example, if I have an ordinary random walk at then
Something like a Levy-Smirnov distribution or something.
for example, you mentioned I'm on a ladder, I start walking,
Yeah.
but I could, with some probability I'm going in a wrong direction. Is resetting ever going to help me in that situation?
I know that in one dimension it does. My understanding is. So the particular statement, this is from Majumdar and Evans, a result from some years ago, just that if you have a random walker in 1D and you're asking, will I ever get to a point on the line? Without resetting, the mean first passage time is infinite. With resetting, it's finite. Just because you don't go the other direction. Go on, in what sense?
Well, I think about
Oh, yes. Yes. But it could be that it's some parts of that space. No. But you are going in a direction that's slower. So I think what you do need for this is that in this high dimensional space, there are some paths that are slower than others. That's the equivalent of the wrong direction. No such thing, of course, here, but yeah. How much slower? I don't have an answer for you yet.
How universal is it for the DNA replication to be the bottleneck for?
Certainly not in anything larger than a bacterium.
So with the explanation for error-correcting codes and like human, for instance, speech, because it's left over.
Well, no, I think in multicellular creatures, there's a great reason not to have a mutation rate that's very high. Well, aside from the fact we have large genomes, the other one is you want to avoid somatic mutations. You want to avoid cancer, and that's a fairly strong selection pressure. I think it's more interesting when you're thinking about unicellular systems.
Interpretation of the sample example, it does matter whether you think it's more surprising that the dome holds up or that somebody made the name with paralyzed. The claim is
Yes.
No, this isn't functional at all because it doesn't have anything to do with
Yes.
But of course, what it does have to do is holding up the structure.
Yes.
Which maybe was much harder than taking the angel.
Oh.
Right? You can take the angel on a wall or on a rock, right? 25,000 years before.
Certainly could. It doesn't make the angel possible. It's just a particularly harmonious place to park the angel. But I think I see your point. You're saying it's really.
It has to do with diagnosing what was the hard part.
Correct. Exactly. No, that's exactly right. And I think that's no. Oh, no, not at all. Exactly. Precisely. So I'm not saying kinetic proofreading is not functional. I'm saying it doesn't have to be selected because it gives you fidelity. Exactly right.
Hi, my name is Maria. I'm a graduate student in Pankaj Mehta's group at Boston University, and I'll today talk to you about these order parameters that we've developed for tracking cell type transitions. So a general overview of the talk. First, I'll talk a little bit about what a cell type is to give you some background. I'll talk about the current standard way of identifying cell type, and then finally I'll end with the work that we've been doing to find some order parameters for characterizing these cell type transitions. So to start, very simplified view of development. One of the basic questions in development is, how do you get from one single cell to many different highly specialized cells all working together in a highly complicated organism? And a very simple answer to this, or a very simple start to the answer to this, is that as the organism grows, the cells divide and multiply, and through complicated signaling processes, as well as other environmental factors like mechanical stresses and chemical reactions and things like that, you end up with some genes being turned on or off. So some genes are being expressed, some genes are not being expressed, some are more accessible, some are less accessible. And because gene expression eventually leads to determining which proteins are being transcribed or translated, gene expression ends up determining the function of the cell. So, depending on the particular gene expression, you might have a stomach cell type or a lung cell type or one of many other cell types. So cell type just generally describes the overall behavior of a cell. And throughout this talk, I'll be referring to basically all of the data I'll be showing comes from mouse lung data. The main reason for this is because our collaborators at the BU Center for Regenerative Medicine are working with mouse lungs. So to give you just a sense of what this system is, as the mouse is going through the stages of embryonic development, in the very early stages, you have a very general lung progenitor cell type. And then as the embryo develops, you end up with more and more highly specialized cell types. And eventually, you end up with these alveolar cell types, which are important for gas exchange in the lung. And so you'll be hearing a lot about alveolar type 1, which are AT1 cells, and alveolar type 2, which are AT2 cells. So one of the really interesting things about development is that specification of these cell types is very robust, which has led to the idea that these cell types can be treated as attractors in a dynamical system. And this was sort of popularized by C.H. Waddington. This is known as the Waddington landscape. It's a sort of metaphor for describing cell types as attractors. Basically, you have cells rolling through these valleys until they end up in these basins of attraction, which correspond to your cell types. But we also know that genes interact with each other in really complicated networks known as gene regulatory networks. Here you have one very small example of a gene regulatory network in mouse embryonic stem cells. And this is just one little snippet. You can imagine that these gene regulatory networks and gene interaction networks can get very complicated. But we know that cell types sort of act like attractors. We know that genes interact in complicated networks. So we can model cell types as attractors in networks. And this is where we have the idea of an attractor network as a model for understanding these cell types and development and cell specification and so on. So now that I've told you a little bit about what a cell type is and how we can think of cell types, how we can model them as attractor networks, I'll tell you a little bit about how one can experimentally identify cell type and the sort of current methods that biologists use to experimentally determine cell type. So here's the central dogma of molecular biology. DNA, depending on which genes are being expressed or not expressed or on or off, basically what's accessible or not accessible, will be transcribed into RNA, and then this RNA will be translated into proteins, and these proteins determine the basic functioning of the cell, as I mentioned before. So if you want to probe what genes are being expressed, you can take a look at the RNA, and this is done nowadays through a method called single-cell RNA sequencing. So you take individual cells and you digest them. You take out these individual RNA and you match RNA to the genes that they were transcribed from. And you end up with rows that correspond to genes and cells that correspond to columns. So each of these little bins here counts the number of RNA counts corresponding to that gene in that particular cell. And something that you might notice from looking at this 2D histogram of RNA counts is that there are a lot of black bins, and these black bins correspond to 0 counts. which sort of brings us to some of the issues or challenges when analyzing single cell RNA sequencing data. So it's super noisy and also super, it's really difficult to actually amplify these RNA in order to sequence them. So you end up with a lot of dropouts, which are just 0 boxes. So there are tons of boxes that have just no information in them at all. Another thing is that if you're trying to identify different cell types, these cell types, especially cell types within organs, like within the same organ, are very highly correlated. So this is showing you the overlap matrix between cell types in, like, these are all cell types in the lung, and the lighter the boxes, the higher the overlap, and you can see that each of these has a very high overlap between these different cell types, which makes it more challenging to identify a particular sample and correspond to the cell type. So now that I've told you about single cell RNA sequencing, how do you actually analyze single cell RNA sequencing data? So this is the sort of standard method for doing it. This is what most biology papers will do. The first step is generally applying some dimensional reduction technique to this very high dimensional, you have tons of genes, so you're taking all of these genes and reducing them to just 2 dimensions using something like UMAP or t-SNE. So each point here corresponds to a different cell. And you've reduced it down to these 2 dimensions. Yeah, here, by the way, if you're interested, I've applied to UMAP. And this next step after you've applied this dimensional reduction algorithm is to use these known marker genes that have been experimentally determined to correspond to different cell types. So it's well known that SFTPC corresponds to an alveolar type 2 cell type. So if we color each of these cells according to their expression of SFTPC, we can see that this cluster in particular has very high SFTPC expression. And so we label that as, okay, that cluster is most likely alveolar type 2. And then you just repeat these for all of the different cell types and all of your known marker genes. And there are certainly cases where this yields fairly reasonable results. So on the left-hand side here, you have, well, both of these are from the mouse cell atlas. On the left-hand side, you have, the lung data, and it's very similar to the lung data I've just shown you, but this is, all of the different lung types, and they've been able to cluster these fairly well. But then there are also many cases where this approach can lead to, results that are really hard to interpret, like all of these clusters are overlapping. Are these truly different cell types, or is this just the reduction of, or the result of dimensional reduction and clustering gone wrong? So overall, the message is, although dimensional reduction can be really good for certain cases, it's an unsupervised method, so you don't need anything to just, throw a dimensional reduction or clustering on there. It's great for getting like a general sense and, visualizing your data overall, but it's not great for getting like a quantitative rigorous measurement that's the same between data sets, because depending on what data you include, you'll get totally different axes. You also get the distances between cells, the distances between clusters aren't very meaningful. So here's an example where this is the original data. It's in 2 dimensions just as to demonstrate the method. And you can see that orange and blue clusters are very close together, but in each of these UMAPs, the distances are the same basically between the 3. So you can't really trust the distances when you're looking at a dimensional reduction like UMAP or t-SNE. And also, the distances aren't consistent also, like between runs of the algorithm. Another thing that would be nice to fix is the fact that these axes don't have a very clear interpretation. It's just, UMAP axis 1, UMAP axis 2, pretty hard to interpret that. And then finally, you have to choose some sort of hyperparameter to, you have to choose which one of these plots to go with, and that inevitably introduces at least a little bit of bias. So how can we use this idea of attractor networks where cell types act as attractors in a dynamical system? As well as this, there's a huge amount of data being created through these single cell RNA sequencing atlases, where they're atlases sequencing organs and tissues across organisms and all of these different systems. How can we combine these things to sort of create a better way of identifying cell types, measuring cell fate transitions, and so on? So our group has come up with these attractor network order parameters, which are a direct application of these Cantor and Sampolinsky order parameters. So if you know about the original Hopfield model, the order parameters in that model are basically the overlap between your attractors and the current state of your system. It's just a dot product. And in the original model, it's assumed that all of these cell types are orthogonal to one another, but we know for sure that that's not the case in the, when we're talking about cell types. So instead, we use this decorrelated Hopfield model, which again comes from Cantor and Sampolinsky. And this is basically you have this original Hopfield model order parameters, but then you're also taking the inverse of the correlation matrix here. Xi here is just a reference basis of your known cell types. So, this is basically saying, here are the gene expression profiles of all of the cell types that I know about in the system. Let me compare, basically take the overlap, but then also take the decorrelated overlap so that we can distinguish these more clearly. And one way you can visualize this is that we're transforming from gene expression space to cell type space. So we start out in this very high dimensional, this is just showing you in 3 dimensions for the sake of simplicity. And you have some sample, you have some known cell type B and known cell type A. And this method is basically decomposing this sample into, doing a vector decomposition onto these non-orthogonal cell types, which is just a projection onto a non-orthogonal subspace. And you end up basically with your sample projected onto these cell types and coordinates in this new space of cell types. So we've developed a Python package to do this for you. You can find it at this GitHub repository if you're interested in that. Yes.
It's like by decorrelating, we'll basically make it so that if 2 cells have very close gene expression, it will still be considered just as far apart as if you have very different gene expression. So it seems like it's kind of, is that a problem that you sort of don't clump things together that maybe should be clumped together, or?
It depends on what you're trying to do. For example, if you want to tell organs apart from one another, then you can just change your reference basis to reflect that level of resolution. So your reference basis can be on the level of organs rather than on the level of individual cell types. And I've done that in this preprint, and you can read more about that there. But basically, this method does work at different levels of resolution, so you can use it for that as well. Yeah, so you can check out this GitHub repo if you're interested. And yeah, so basically now we're going from this difficult to interpret set of axes that we get from UMAP to these more easily interpretable axes, which are, whatever cell type 1, cell type 2. And you also get a more quantitative measure because, as I said before, the distances between these clusters is not meaningful. But in this system, it is meaningful because you can plot as many data sets as you want on this. And as long as the reference basis is the same, you won't get weird things happening with your cluster distances changing and that sort of thing. So now I'm using these order parameters. So I'm using the order parameter that corresponds to alignment with alveolar type 1 for the x-axis here, and the order parameter corresponding to alveolar type 2 for the y-axis here. And the way that you can interpret this graph or this plot, is that these cells over here are most like alveolar type 1 cells. They have high alignment. These are most like alveolar type 2 cells. And then these over here look the least like alveolar type 1 or alveolar type 2. And we can compare these with the labels that we got from before from using marker genes and clusters. And we can see that it agrees quite well. So, the alveolar type 1, which are the blue circles, look most like the alveolar type 1 on our axes. These look most like alveolar type 2 and so on. But something cool that we can do, now we can actually compare data across from time series. We can compare them from different sources. We have axes on which we can directly compare these different things. So here we have mouse embryonic data. These are embryonic mouse lungs that are going from embryonic day 12.5 up until postnatal day 42. So from early on in embryonic development up until 42 days after the mouse has been born. And you can see that the, in the very earliest days, it starts off looking not much like alveolar type 1 or alveolar type 2. And then later on, you end up in these basins of attraction, as one might expect. And here I'm showing you the same information, just frame by frame. And you can see that, not only do we end up with these basins of attraction as we expect, we also have this weird sort of in-between phase that cells seem to be passing through or getting stuck in. And then when the mouse starts breathing between this day and this day, it seems like that slowly gets destabilized over time. So yes.
Oh, are these cells the same from frame to frame, or are they just from like the same, like?
They're from the same data set, but these are different time points. So it's like all mouse lungs and all alveolar type 1 and alveolar type 2 in that are being developed just on different days of development.
If you track the same cell over different days of development, or?
You can't do that because single cell RNA sequencing destroys the cell.
Okay.
So it would be great to do that, yes.
Like, there is no at non. There's the other cells, but something else. Like
Oh, and at 0, 0. Yeah. So in the other ones, I was including cells of other cell types in the data set. Oh, these are all, I just take the labels from this Zepp et al. Paper. So they used some like marker genes for progenitors of alveolar type 1 and alveolar type 2 cells.
the other question I have is about scale.
Okay, yeah.
Because projection of 24 it isn't doing very much like it's the most you see, but is that not very much?
Yeah, that's a good question. So yeah, so the maximum that you could get with this method is like basically ranging from -1 to +1. So you would expect like if it aligns perfectly, we get 1 every time. But again, this data is like mostly zeros. So if you aggregate them and like take the average over all of these cell types, you'll get something more like 0.7, like you'll get a lot closer to 1. But with these individual cells, like most of the genes are just 0. So yeah, the scores aren't very high, but I can, if you're interested, I can show you some histograms where if you project a case where the sample type doesn't match the type you're projecting on, the scores are always between like a very small area around 0, basically. So it's really hard to get a high projection when you don't actually have look like that type. I don't have a good way of checking that, but yeah, I think I would say it's probably mostly biological noise. I mean, mostly issues with measuring, because like I said before, it's really hard to amplify this RNA. And so it's really hard to measure it in the first place. But as for how much like inherent heterogeneity there are between these cell types, that's definitely a question that I'm interested in exploring more, because there are certain cell types that are a lot more heterogeneous than others. Yeah. Okay, so, oh, yes. Yeah, so in the order parameters, it's kind of assumed that, in the very basic Hopfield model with no fancy potentials or anything like that, it's kind of assumed that they're all just like equally deep, like well potentials, basically, like parabolic potentials. But yeah, I think that basically what I'm working on nowadays is So this is just like trying to see what order parameters can tell us about the data. But what I'm working on nowadays is sort of looking at different potentials and looking at the changing landscape over time and looking at like, okay, how can we build in a potential that destabilizes over time, and you have, you know, a saddle node bifurcation and that sort of thing. So, does that answer your question? Okay, any more questions? No, cool. All right. So here's the, you know, same animation that I was just showing you. And, you know, we can model these, you know, we have an attractor network, we can just simulate an attractor network. So one thing that you can do with this is simulate an attractor network with a shared progenitor between these two final cell types, as well as one without. And you can see how the order parameters change over time. And so it seems like this sort of behavior is suggestive of a you know, a shared progenitor state, because these cells are going off into the middle and then splitting off. And we know biologically in alveolar lung types, this very much is the case. So perhaps we can use, like, by looking at these order parameters across different cell types, they can be used to sort of reconstruct the lineage trees. And we're sort of working on that in C. elegans and trying to reconstruct the C. elegans lineage tree using this method. So we'll see if it works. Yes.
Is there any like seeing how much overlap there is with the BP AT1 to like pre-step that you have and the actual AT1 and 2? So like could like that point before it splits be actually that precursor to your cell, or is that like
the point?
You know, so like seeing that bottom left, you have that like precursor cell. Like,
oh yeah, right.
So like is that one like that thing before it splits?
That's what I would suspect.
Okay.
It's we would have to you know, ask our collaborators to do experiments to like check the stability of that sort of thing, because as I was saying before, like, so I haven't gone much into talking about the potentials and the actual landscape, because in the basic Hopfield model, it's, you know, it's all very simple, but in development, it's actually quite complicated, and depending, like, your landscape will be determined like directly by what signals you're receiving, what state in development you are. So in theory, if you pause development at this stage, it's possible that they can get stuck in that in-between state, and that could be one way to check the stability of that particular state. Oh yeah, no, I think I said everything I wanted to say about the slide. Yeah, so you can sort of check whether or not it has a shared progenitor, and you can perhaps reconstruct these lineage trees. We're working on testing whether or not this will actually work. Yeah, so basically, to summarize, attractor network-inspired order parameters can give you intuitive coordinates for analyzing this single cell RNA sequencing data. So not only can you use it to identify these cells and give you sort of a quantitative measure of how like these different cells is, rather than just doing a dimensional reduction and clustering, you can also measure time series data and measure day-by-day transitions. Something that I didn't talk about here, but something else you can do, since you can compare between different data sets. If you look in our preprint, you can find that we compared between these engineered in vitro cells with these in vivo endogenous cells to tell how much these engineered cells actually look like cells that come from the real mouse. And then finally, perhaps we can also predict some lineage trees. So I'd like to thank my group. I'd like to thank our collaborators and our funding sources. If you are interested in reading more, you can check out our GitHub repo, or it's also on the Python package index. You can also check out either of these preprints for more info, and thank you for listening. It's definitely something I'm interested in looking more into, but most of the time-consuming part of analyzing this data is sort of pre-processing it and getting it into a nice format. So looking across all of these different organisms and getting all of them into a nice format, while it would be awesome, it would be also disproportionately time-consuming. So, yeah. Yes. So the time steps between these are quite large. I would expect that like different cells are in different stages of this differentiation. So this is looking at, like, the simulation is looking at like
assuming that they're all exactly coordinated perfectly, going like differentiating at the exact same time, whereas in reality, we know that this isn't the case. We know that different things will be at different time stages. So these time steps are pretty wide, so it'd be hard to tell from this data, but there is a data set that recently came out that I'm interested in looking at that does sort of go at a higher resolution of time by time, day by day.
Looks like the group that is taking longer, but there is also a main group that is pretty immediate.
Right. It's also somewhat hard to tell, like, it might look like these cells, these cells went directly up here, but they also it might be going through this process. It's just we haven't looked at every single time step in between, and we're just looking at cells in all different stages of this differentiation. Yes.
I'm just curious, I understand the age are very close, but if it didn't have a colleague who had labeled those, could you have worked to that by, do they appear as somewhat separate clusters in UMAP?
Yeah, they definitely appear as separate clusters, yes.
So then you actually need someone to label them for you, right? You could have taken those clusters, find the centroid, and then your approach on those centroids.
If I know all of the marker genes and that sort of, like if I cluster them myself and that sort of thing, is that what you mean?
Clustering just by UMAP, if no one has told you anything about the biology, just have single cell data.
Yeah, I could probably guess like, okay, I'm just going to assume that these different clusters are different cell types and then construct my bases. Yeah, I wouldn't know what to label them though. Yeah. Well, what about secretory and basal and such? Yeah. Go ahead. So the order parameters come from the reference basis. So are you asking, how can you find the reference basis from scratch, like without anything else?
A little bit like that question. If I wanted to do this in another way, how would I do it?
Yeah, so I think in order to get the reference basis in the first place, you probably would have to do some dimensional reduction thing and clustering and that sort of thing. So I think where this is really useful is looking at samples where you have, you're either engineering a cell type or you want to, the end states, but you don't know what happens before that. You don't know what happens in between, or you have some in-between state that you want to study more. So yeah, if you wanted if you were looking at a data set where you had no idea what the cell types were, you would need to do some UMAP and dimensional reduction and that sort of thing. Yeah. So does that answer your question? In the mapping, okay. Do you mean this mapping of applying the decorrelated
It was like
thing?
a picture where you went from it's like you had these two blue graphs, and one went from so maybe that is exactly that matrix,
Yeah.
but what I guess is there some information between that you could learn something about your system?
Yeah. So you mean the actual projection that takes you from this space to this? Yeah. So that's actually a matrix that's sort of we call it the predictivity. It basically tells you which genes are the most predictive of which cell types. So this is actually a way that you can sort of get marker genes out of this method. So if you clustered something and you felt like these cell types were separate enough and you found the predictivity matrix, you could sort of determine these are the top genes that are responsible for these cell types. And when we do this, we find, yes, indeed, SFTPC is a marker gene of alveolar type 2 cells. So, do you mean well, so they end up at these 0, 1, and 1, 0, right? So these are 0, 1, and 1, 0 are the attractors of the system. So are you saying do they go farther this way, or I guess I'm not sure what your question.
Oh yeah, no, I guess I wasn't clear with the fact that they were the attractors.
Okay, yeah. So these these are these coordinates correspond to the attractors because they correspond to basically when you've decomposed these vectors, 100% of the decomposition goes to one vector. So it's just, 0, 1, and 1, 0. Yeah.
So is there any like change in the gene expressions after they separate?
Yeah. So this corresponds to, like, as it's changing over time, this corresponds to changing gene expression. So in very early times, you have a very embryonic stem cell looking gene expression profile, and then in between, we find this in this in between cluster as well. It looks like an exact mix of, like, half the genes look like alveolar type 1, half the genes look like alveolar type 2. And then we know that there are certain pathways responsible for determining alveolar type 1 and alveolar type 2. So, for example, like the hippo pathway. So if the hippo pathway at this point is like turned on, then it will go one way versus the other. Does that answer your question?
So hi, as you said, I'm Christine. I am coming from the Paris Brain Institute, more specifically the Wired Lab. And today I'm going to present and discuss how we improve the traditional Granger causality analysis to infer information flow from calcium imaging data, information flow between nodes. So a major challenge in neuroscience is to understand how information flow between neurons in order to trigger specific behavior. And we really want to move beyond the simple identification of which neurons are active toward the characterization of functional circuits and to reconstruct causality networks that describe how neurons influence each other. So to do this, we take advantage of the larval zebrafish model and its genetic accessibility, its transparency, and its small size. So it's a vertebrate species, which means that it has the same, well, not exactly the same, but similar brain regions as other vertebrates, especially those that are involved in locomotion, which I am studying. And we can create some mutants that have fluorescent neurons. So we can record these neurons in vivo because the larval zebrafish is transparent. And since the brain is really small, about 100,000 neurons, we can record all neurons simultaneously. And also for lab, it's much easier and cheap to maintain than mice and primates. So calcium imaging, I don't know if you're all familiar with it, but it's a type of microscopy used to record the fluorescent activity of the neuron. So the neuron have a calcium indicator, and when a cell is active, a neuron is active, there's an influx of calcium, and this binds to the calcium sensors and make the neuron become fluorescent. So in this data set, we have recorded the neuron from the hindbrain, so it's a part of the brain involved in locomotion. And here you can see some example fluorescence traces, also called DFF. So now we can use these traces to apply Granger causality on it and try to infer information flow between neurons. So Granger causality originally comes from econometry, and it's very commonly used also in neuroscience, especially with EEG data, to look at the influence of one brain region over another. But it's more recently being used to look at the cell resolution, so the neuronal resolution. And unlike correlation, it's a directed measure. Correlation really identifies pairs or groups of neurons that are active at the same time. So that means that either one neuron drives the other, or one region drives the other, or that one common input drives the two neurons, but it doesn't give the directionality. By contrast, Granger causality is a directed measure. So it really says which neuron influences which neuron, but it does not mean that there's an anatomical connection between the two neurons. So the core idea is that if we can use X1, the time series of event X1 to predict the past of the time series of event X1 to predict X2, then it means that there's an information flow from x1 to x2. So this assumes that we can model the two time series by vector to regressive models. And so there's a reduced model, which only takes the inputs as input the past time points of x2 to predict the future time points of x2. And there's a model in which to predict x2, we use both the past of x2 and x1. And then we compute a p-value and an f-test, and we compare the variance of the prediction error, and if so it's W2 and U2. And if W2 is its prediction, its variance is smaller than for U2, then we say that X1 Granger causes X2. So then in multi-component systems, we also want to take into account the other variables. So we change the vector to regressive models to incorporate these other variables, but otherwise the expression for the Granger causality value and the f-test are the same with the number of parameters adjusted. And this gets much more computationally expensive when you get to hundreds and hundreds of neurons. So first we simulated synthetic data to verify that we could use Granger causality to retrieve the true underlying structure. So we used a small networks of 10 neurons. The arrows in the network and the red dots in the connectivity matrix represent excitatory links, and the short bars and the blue dots are inhibitory links. Then for the first simulation we did, we used a simple vector autoregressive model. So it's the same that underlies the Granger causality analysis to mimic the neuron's activity. And we compute the correlation, the bivariate Granger causality, and the multivariate Granger causality. And we can see that the correlation really misses the underlying structure of the network, and that the multivariate Granger, so the black squares represent a significant link found, and the dots are the true links. And we can see that multivariate Granger causality retrieves successfully the full underlying structure, but the bivariate Granger causality has some spurious links because it cannot detect between direct and indirect links. So for example, neuron 1 to, I don't know, 5 or 6, it will consider that there's a path to it. Oh yeah, something I forgot to mention in the definition of Granger causality is that the parameter P here is the number of past time points that we take into account for the prediction. So for example, it's what we call the time lag. For a lag of 3, we're going to take into account the 3 previous time points. And yes, there is indeed, some can be 0, some can be positive or negative. But in the end, we just look at this value and we call that the GC strength of the link.
So if you're to continue to increase P, that's sort of a thresholding, and eventually you may extract the actual connectivity because it would only be
yes, but you don't necessarily have only one time steps that can count for the connection. It can be like integrated somehow over several time steps. And also what I'm going to show is that I'm using calcium imaging, so calcium transient, and the dynamics are really slow compared to the real spiking activity of neurons, but I'm going to come to that. Yeah, so in this simulated data, we use the P of 2, the order 2. So we can have, well, it's only up to 2. So we also looked at the cross correlation, and well, I have additional figures if you want to see, but this is just an order of 2. Can you repeat your question? No, 2 time steps. Yeah, so when I'm gonna come to the real data, this is simulated data. It's only time steps. We took an order of 3 because we looked at the difference between taking like 4 or 5 time steps, and it was almost the same network. So we kept the simplest possible, and it's comparable to the known. Well, I'm gonna come back to it. If I forget, remind me. But then we tried to mimic something more realistic for spikes. So we used a generalized linear model, and the spikes were generated with a standard Poisson process. And again, we can see that the correlation misses the structure, that the bivariate Granger causality has many spurious links, and that the multivariate Granger causality retrieves the true network. And for our last simulation, we convolved the spikes with an exponential decay function to be able to mimic the calcium signals observed in real neurons. And this time, so correlation still misses, bivariate still has extra links, but multivariate Granger causality also has a few extra links because of the similar calcium decay that neurons have. But it still performs better overall. So we validated the Granger causality on synthetic data, and now I'm going to talk more about biological data. So we have this data set recorded in the hindbrain, and we categorize the neuron into 3 categories. The medial motor correlated neurons, so those that were correlated to the activity of the fish. The other motor correlated, so in red, the other motor correlated neurons in blue, and the non-motor correlated neurons in gray. And for some biological reason, the most interesting to us were the red neurons. So we first focused our analysis on this one, and we applied the Granger causality on them. And what we obtained is an almost fully connected network. So we know that it's biologically not very plausible. So we decided to try to customize the Granger causality method to calcium imaging data using another data set in which we know the true information flow, because in this one, we don't know what the actual information flow is. So this is the motor neuron data set. So it was recorded in embryonic zebrafish. So motor neurons are the neurons in the spinal cord that make the fish move. We know the expected network of information flow that should go from the head to the tail on either side. So we can reconstruct this expected network. But when we apply Granger causality, we see that we really don't have this expected network. So we're creating a pipeline to, we created a pipeline to make it better. Today, I'm just going to focus on this part because these are other steps. So what happens a lot with calcium imaging of live zebrafish is that we can have some motion artifacts, either because the fish moved a little bit, so the brain moved a little bit, or because of the setup that also moved a little bit. And so in this data set of 11 neurons, I think, we have out of 1,000 time points recorded at 4 Hertz, so 1 point every 250 milliseconds. We have one single time point for all neurons that there's a drop in fluorescence that is not, that is an artifact. And so with this artifact, we see that this neuron, number 10, really takes it all over, and it looks like he's driving all the other neurons. But once we correct this motion artifact, so we just interpolate, well, here it's just the average because there's just 1 point, but if the motion artifact is longer, we interpolate it. We see that we get rid of this super driving neuron and it's still not close to what we expect, but it's already better. And we looked a little bit at why it was this neuron that was taking all the drive for, and it's because he's the neuron with the lowest activity, so it has a lowest signal to noise ratio, so the single time point is perceived as having a big effect on because the rest of the effect is no. Noise is a big problem, as we saw earlier, for Granger causality, but another problem is correlated noise. And in this recording, it was done with a microscope called spinning disk, so it captures image by image, and the noise is correlated across pixel. So we have to smooth the signal, and we use a method called total variation regularization. If you want to know more about it, you can have a look at this paper. And just in contrast to, for example, low-pass filter, it really keeps the fast rise of the calcium signals, and it doesn't shift the start of the rise, things like this. So this is on unsmoothed data, and this is a result on smooth data. Same here. And we can see that we get rid of many spurious links from right to left and left to right. Another thing that we still have many spurious connections, so we wanted to look at if we could really use the F-test that is traditionally used in the Granger causality. So to do that, we broke the causality between two time series by shifting one of the two signals. And computed the F statistic on the shuffled data. So it's the one in green. And we can see that compared to the naive F in light gray, the fitted shuffle F has a shifted support. So that means that if we keep the original threshold, we will classify many links as being significant, even though they are not. And here we have a lot of outliers because of the pseudo periodicity in the signal, because the fish is swimming. Yes. No, it's head embedded. It's, oh yeah. So it's, yeah, I'm sorry, I forgot to mention it. The fish has its head embedded in agarose, so we can like record the brain, image the brain, but its tail is free, so we can also record its behavior at the same time.
I guess it has motion, like there's no way to know what the head would be.
What's
like, because I just think of the fish swimming, the head would be moving too.
No, it's embedded in, it doesn't move. It's like stuck in agar. It's a bit like I'm holding your head and yeah. Head fixed, yeah. So we compute new thresholds we parameterize this curve, this new F distribution, and then we recompute the Granger causality on this. And we can see that we get rid of all the spurious right to left and left to right connections. We did the same for the multivariate Granger causality, and we also get rid of these connections that are supposed to be only direct because we are conditioning on the other neurons. So we did that for the 9 fish recording that we had. So it's very much improved. We don't have any more right to left and left to right connection, but one big problem that we have is that we still have tail to head connection that should not be there. And that's because there are 2 reasons. First, the recording frequency, this is an old data set, it's from 2012. And the frequency it was acquired at is 4 Hertz, so 250 milliseconds. And it's quite slow compared to the real spiking activity. And also it's using an old calcium indicator So it's really slow. The decaying activity is really slow after a neuron has spiked. So with higher frequency recording and newer custom indicators, we could have better results. So then we went back to the hindbrain data sets, and we have a much more sparse network. We computed the new threshold a bit differently here because we noticed that the F distribution of the shuffled signals was just a constantly scaled F distribution, and for each pair of neurons. So now we have one threshold specific to each pair of neurons. We did the same for the bivariate Granger causality. Here, the F distribution really matches almost perfectly. But we have a very, very sparse network. And this is probably due to the fact that we're using motor correlated networks. So obviously, there's a strong correlation between the signals, and only the strongest links remain. But we still looked at the distribution of the angles of those links. And we noticed that there was a biased right to left information flow, which was consistent with a majority of left band of the fish tail. So probably these command neurons will recruit left motor neurons in the spinal cord, because these are the hindbrain neurons. So then we extended a bit our analysis to all the motor correlate. Yes. Sorry. I'm close to the end. To all the motor. This is just an example of how it can be useful for biology. So maybe you're less interested, but we found something cool. So still going to talk about it quickly. So we extended it to all the other motor correlated neurons, not only the medial ones. And again, we had like mostly left angles because the stimulus was coming from the right of the fish, which makes it move to the left. And the GC links also gave an information flow mostly from the right to the left. And there's a high motor center that has been identified from functional and anatomical studies in many vertebrate species, including the mice, the cat, and I don't remember which ones. And it had been identified in the larval zebrafish last year. And it's called the it's called the mesencephalic locomotor region. And it's really known to be like you stimulate this region and the animal starts to move. And what we found is that we had very strong driving neurons in this region that is known to drive neurons that are closer to the tail, and other neurons in the left mesencephalic locomotor region. So we can see it on other planes of the same fish. And this is, to our knowledge, the first time that was shown that using the activity that we could determine this region. So we thought that was a super cool result that was not expected. We were not looking for it. And so to conclude, Granger causality is an effective method to retrieve information flow, but we have to take care of the non-linearity, non-gaussianality, and the noise when using calcium imaging data. Our pipeline consists of both preprocessing the calcium traces and adapting the threshold for the significance of the Granger causality links. And because the neurons' calcium activity is similar across species, it's not limited to the larval zebrafish. And for now, we suggest that because multivariate Granger causality is really prone to overfitting, when you have a lot of neurons, you have a lot of variables. We don't really suggest using it on large data sets. And so this is our paper that came out in February, if you're interested. And yes, thank you for your attention. Thank you. So I just want to thank the postdoc that was working with me, who's a real biophysicist, because I'm not a biophysicist myself. And my two thesis advisors and other collaborators who work on the project and my lab and my dog. Do you have any questions?
You had this on the conclusion slide, but what would be the difference in, I guess, like the pipeline between these for large number of neurons and
So bivariate is really looking at a single pair of neurons, ignoring all the rest. And multivariate range of causality, you look at the influence of one neuron on the other, but conditioned on all the rest of the data sets that you have. And so the rest is the same. Thank you. Yes.
So if you count for the situation, because if there are two independent neurons that are similar with single biophysics, and they oscillate at the same frequency, they would automatically be correlated, right? Even though they are not connected at all. Could be not connected.
So we don't really have oscillating neurons in this case. We have one problem with multivariate granular causality is the redundancy in the signals, because the brain is really redundant and you have a lot of neurons doing the same thing, even though they're not really connected. And that's why we say that it's not a direct anatomical connection. It can like give you suggestions on for what to test then electrically, for example, with backfilling, things like that. But we don't have neurons spontaneously oscillating at the same frequency. I think it's, I'm not really good with human brain, but I think it's more in like the cortex and stuff like this, maybe. No? No, it's not periodic. Yeah, wait, I can show you. Go back to the. So on this slide, for example, this one is periodic, and it's a neuron that is responding to the visual stimulus that is periodic. The visual stimulus is like a grading passing by every 5 seconds or something. But we don't look at these neurons. We look at those other neurons that it looks a little bit periodic because it's when the fish moves, so when the fish sees its grading. And yeah, that's also the reason why we have a lot of spurious links in the bivariate granular causality. But when we shuffle the data, when we time shift it, it should be disrupting this causality.
Okay, yeah, thanks for having me. So this talk is about sequences and thinking about sequences in naturalistic behavior. So we start with the observation that animal behaviors occur through time. And one way to model this is as a transition between discrete states. Okay, so we think of a bunch of these discrete states and we're transitioning between them. So I have this, picture of a running horse just to kind of demonstrate. Now, these states and the transitions between them could be generated by networks of neurons that drive the behavior. Right? That's natural to think of. And so there are more concrete, analogs of these discrete states in certain cases. Like some people think that each state could drive like a motor motif, right? So you might say, okay, it's a little bit weird that these states are discretized, but actually, if you have a discrete state like state 1, that could drive a whole continuous little motor motif. And there are also models that have been very successful that also make this assumption of discretization of the states. So in particular, SynFire chain models of birdsong learning and birdsong generation. So here we have kind of these discrete chains of basically collections of neurons, and each collection will fire at a particular time, which corresponds to a time point in the song. Okay, and if I have time at the end, I will talk about the possibility of relaxing this assumption, but perhaps the best thing about this assumption is that it makes it easier to model mathematically. Okay, now these models of thinking about sequences that go from state to state like this are particularly applicable when we're thinking of memorized sequential behavior, or things that are almost, you don't really have to think about them because one state leads to the next automatically with little or no external control signal, right? So if I start in state 1, I will just progress to 2, etc. So maybe things like walking or something, these kind of behaviors that are almost automatic. Yeah, or like a marble rolling down a hill. Once it starts, it keeps going. Okay. So, now we ask the question of how is it that sequential behavior could be learned in the brain? Okay, so we kind of basically use this framework that's discussed in this paper as our framework, which basically posits that there's a tutor signal that may be allowing these memories to be formed, right? And this paper suggests that motor cortex is actually sending this tutor signal to subcortical motor circuits. So how would this work? You'd have motor cortex, which sort of serves as a general purpose controller. So it's basically like you're thinking about your movements. And you can maybe use the analogy of learning a tennis swing, that at first you really have to think through the whole process. So the motor cortex is driving it. But at the same time, motor cortex is sending signals to subcortex. And this could be like a tutor signal, so that eventually you don't have to think about it anymore. Motor cortex no longer has to explicitly drive the behavior, and it becomes kind of automatic, just something routine. And those automatic routine behaviors could be driven by subcortical structures. But you need to figure out a way to get, basically the action into subcortex. Now, you could also think of the tutor signal as just being provided by other brain regions. You could think of it as being provided by the sensory signals themselves. That's not so important, but it's just this idea that there is a tutor signal which allows memories to form. And this paper also, basically points out that, okay, there are some behaviors like dexterous movements that almost always involve, deliberate thought. And in fact, experiments show that motor cortex is important for these kinds of movements. But there are also movements like basic movements, or movements that have been like overtrained where you don't need motor cortex. So that's kind of the evidence that these subcortical structures are not only driving kind of innate behaviors, but also overtrained behaviors, things that have been learned to the point where they're almost automatic for the animals. So you can think of sort of a computational framework that looks something like this. And unfortunately, it looks like this kind of blocks that last box. I guess I'll just have to describe what is in there. So, motor cortex serves as this general purpose controller, which allows you to get in the vicinity of a solution quickly. So if you're learning a tennis swing, you will not do very well once you start, but it'll be a lot better than just random motions, right? So you can observe someone else's tennis swing and get close to the actual solution. Then you need some way of incrementing that solution, and that can be a slow process of practice. You have to practice for the 10,000 hours or whatever it is to master the tennis swing or the tennis serve. And then finally, you need some mechanism of storing the memory storage and retrieval, which protects the solution from being overwritten as you continue to learn other things or you have other experiences, right? And you also want to be able to quickly and effortlessly recall the solutions that you have worked so hard to learn. And this talk will primarily focus on this block, which is the memory storage and retrieval part of this sort of trifecta of motor learning, so to speak. Okay. But okay, there's the issue, right? This is like the reason why the project exists, of course. This is the gap in knowledge, which is that it's not currently well understood how tutor signals, these guys, could give rise to memorized sequences. Like, how does that actually work? So that's what we're going to look at. So there are models that are basically situated in this world. And here I've listed a couple of like key papers, but there's a much longer list. So this is not comprehensive, but the basic idea looks like this. So I have, let's say, two patterns here. These are like the states. They correspond to the 1, 2, things that I had previously. So these are the two states. I assume that they're length n. And I have some weight matrix here. I'm going to define it like this. So this is an outer product of these two vectors. So you can, get this by basically doing your typical matrix multiplication of these two vectors. And we have this dynamics here. So this is going to be like the dynamics of a neural network. And this neural network here, this is the connectivity between these neurons is recurrent. Okay, and so we have some nonlinearity phi here. And this phi, basically, it's not so important exactly what it is, just that it looks like this, right? So it's kind of does this binarizing operation. We call this like a sigmoid shape, right? And so why does this, do what we want to do in the context of sequences? Well, if you take W and you just multiply by some random pattern, then basically you get a dot product of this and this random pattern, and that will be approximately 0, because these are, it's assumed that these are high dimensional vectors, right? And then if I multiply by pattern 2, assuming that pattern 1 and pattern 2 are uncorrelated, you also basically get 0. But in the special case where you multiply by pattern 1, you get pattern 1 times pattern 1, and that's a big number. Okay, so this, and then what's left over is this pattern 2 here. So this thing dot products this thing. So that just turns into a scalar, a big scalar. And then you're left with this vector, pattern 2. So you get that this, points in the direction of pattern 2. So what this means is that if I start in pattern 1, I wind up in pattern 2. So this is similar to the argument for standard Hopfield networks. It's just that instead of, in a Hopfield network, this would also be pattern 1. But now we've just replaced this with pattern 2. So instead of going to pattern 1, you are going from pattern 1 to pattern 2. Okay. And so we say that this encodes a transition from pattern 1 to pattern 2. Hopefully that's clear. Feel free to interrupt me if that's confusing. Now, okay, so that's for pattern 1 to pattern 2, but we also want to basically be able to chain together a longer sequence from pattern 1 to pattern 2 to pattern 3, et cetera. So we just add this term to the sum. So this, gets us from pattern 1 to pattern 2. This one gets us from pattern 2 to pattern 3, et cetera. So that's how we build a whole sequence of these guys. And now, we need a way of essentially measuring the neural network activity. So we use this overlap quantity, which was presented a couple of talks back, where basically you take the dot product of the pattern, particular pattern, and the state of the network. And this will tell you how much the current state of the network is in state mu, right? So how much it aligns with the pattern xi mu. Yeah, so we could go through it. So let's say that I had pattern 2 here, xi2, and then I multiply here, so you factor it in. Then xi1 times xi2 will be basically 0. So this would, yeah, so it just picks out the thing that you want. Exactly. Cool. Thanks for asking. I'm glad I have a chance to clarify. Let me just hide this real quick. Okay. Okay. Now, if you plot this, it looks like this. So I've actually left off the overlap with the first pattern because it just decays exponentially from 1. So it's kind of a special case. Here are all the rest of them. So this would be like the overlap with pattern 2, the overlap with pattern 3, And then these vertical lines are the locations of the peaks in time. Okay, so you see that it's basically equally spaced peaks. Now the width of these overlaps does increase, so it spreads out, but the actual locations of these peaks is pretty constant through time. Okay, so each one of these colors, just to be clear, corresponds with a different mu index, which corresponds with a different pattern. Okay. Yeah, so this is through time. So this is like basically shows that you get the sequential activity. Okay, so this model here that I just described arises from a simple Hebbian learning rule, and I'll define that later if you don't know what that means, with a temporal offset of t minus delta t. So it's basically, there's this fixed offset. But the thing is that this model can be generalized, and this would correspond to arbitrary Hebbian learning rules, and in particular, the Hebbian learning rules that we actually see in biology, right? So that's key. So we want to have a model that actually corresponds with what we see in biology, but it's currently not understood how, these sequences with these arbitrary Hebbian learning rules would behave. Okay, so let me go ahead and, let me go ahead and first talk about, what can happen when you have general Hebbian learning rules. So first off, I'm going to build this weight matrix W. So I'm going to start with this component that we're familiar with. This gives us the transition from pattern 1 to pattern 2. But now I have transitions from pattern 2 to pattern 3, et cetera. But I can also have components that look like this. So I could have transitions from pattern 1 to pattern 3, pattern 2 to pattern 4, et cetera. And these terms will arise when we consider general Hebbian learning rules. And we can also have connections from patterns to itself. Sorry. And we can even have connections backwards. So we start in pattern 2, and then we are activating pattern 1. So all these things will arise. And of course, we may want to weight each of these terms in the weight matrix by some coefficient. Here, I'm assuming that the coefficients are the same, basically across different parts of the sequence. So from 1 to 2 is the same from 2 to 3. I'll talk briefly about what happens when you relax that constraint, but that makes the math a lot easier, and it's sort of corresponds to something natural in the learning model. Okay, so, and for convenience, it's nice to okay, well, I also lost that, but basically if I take this W mu, so this will be like a slice through a vertical slice corresponding to, yeah, so this would be like a vertical slice corresponding to a particular mu. So that's going to be useful to define. Okay, so here we go. So let's talk about Hebbian learning now. So we have a spike timing dependent plasticity kernel. Sorry, this is was originally for a neuroscience audience. So STDP is one of those like super common acronyms we have, but it means spike timing dependent plasticity, which uses Hebbian learning. So it means that if I fire at time t and you fire at time t plus delta t, then I'll adjust my connection strength to you by W delta t. And okay, by me and you, I mean, I'm a neuron, you're a neuron. We fire together, we wire together. It's good. Okay, and you can do this for a whole population at once. And the way you do this, okay, so the pattern determines who is firing and who isn't, okay? And so you can do that. Now the tutor signals here set the state of the network to different patterns at different times in the sequence. So here's kind of my time course. I'm moving along through time, and I assume that a tutor signal is coming in, and it's setting the network to this state, then to this state, et cetera. And so those patterns correspond who's firing and who isn't. And if I start from a 0 matrix, then the final connectivity becomes the sum of these W mu's, where each W mu is equal to this thing. Okay, so I probably don't have time to really let this sink in entirely, but the basic idea is you're doing a convolution of this, this would be the STDP kernel or the Hebbian learning rule, if you want. And these are the states of the two neurons, right? So neuron I and neuron J. Now, if I'm at time, so I'm assuming basically here that I'm in this time interval, because this guy here is the length of this time interval. So if I'm in this time interval, then I know that this neuron is in state C mu. So that's where that comes from. But then this guy, so basically the neuron that I'm talking to, or that I'm, my partner neuron basically. So this is, what is firing at time S. So this would be looking along here. So this can change. So if I'm looking at this time offset, so this is t, this is my offset, then it might be the same neurons are firing. But if I look far enough into the future, then it'll be a different set of neurons that are firing than it was at time t. Okay, so basically you just move this guy along, and you do this convolution. And let's just demonstrate with a simpler kernel here, just for so we can get a grasp on it. So if I move this along, what you'll see is that you get these terms. So you get an A0 self-connectivity. So that's everything where you're at time t here, and everything that the s's that are also in this interval. But then you also have some overlap with the next interval. So that will give you this term. And so as you see, if I move this along, then I will be 0, though, by the time I get to this third interval. So that's why I only pick up 2. Okay. Now, if my intervals are delivered faster, then I'll pick up more terms, because this will overlap, this kernel will overlap with more patterns. So then I'll pick up this component here. And that's why I need to consider these sorts of terms in the weight matrix that are not just from me to the next guy, but also to the guy 2 ahead of me. Likewise, I can get, if I have this kind of a learning rule, which is a very commonly seen one in biology, I can get connections to the previous pattern as well. Okay, in a similar way, because this guy, start here, you actually get connections to the previous guy. Okay, and in general, it looks like this. So this would just be a general expression where I, allow any possibilities, any coefficients. And so remember that the dynamics look like this. So this is my network. And I start the system in, the first pattern. And now, so we're interested in these overlaps. So the way that we get a handle on this is we take the n to infinity limit. And for Gaussian patterns, you get these, basically these are the appropriate order parameters for the system. So you get these effective equations that govern the overlaps, where this g of t is a function that captures the nonlinearity of the original dynamics. Now, we can say more than that, but it's just a little bit complicated. So I'm kind of sweeping that under the rug here. Oh, and yeah, so one more thing. It's useful to also consider it when I have noise here. So this would be like a Gaussian noisy vector here. So this would be like noisy neurons. So that'll be interesting to consider as well. Okay, so let's continue. So this is my general equation again. And so now we need one assumption to continue, and that's that this g of t guy tends towards a constant value, which I'm going to call g infinity, over time, right? So there is a limit as t goes to infinity. And basically we can reason that g infinity should be such that this linear system now, right? So now this is a linear system. And we know how linear systems behave, right? They tend to either diverge to infinity, they go to 0, or this is kind of generic behavior, or they're kind of perched right on the edge of stability. So they neither diverge nor disappear to 0. And it turns out that, if we want this system to be a good model for this system, then it can't go to 0 and it can't go to infinity, unless this one goes to 0. But we're assuming that this one won't go to 0 over time. So that means that this has to be perched on the edge of stability. So if you reason this way, you can find the g infinity that actually perches this linear system on the edge of infinity. Okay, and for convenience, I put this bar over the coefficients when I just absorb the g infinity into the coefficients here. Okay, so g infinity should be such that this linear system is on the edge of stability, and this implies that g infinity just equals the inverse sum of all these coefficients. So this will perch the system on the edge of stability, with some assumptions here, which I'm kind of gliding over, but. And there is a catch, which is that g of t, this expression, is actually bounded from above, okay? While this guy is not. So basically, g of t should approach g infinity, which is given by this, unless it can't, because it's bounded, okay? I'll illustrate that in a little bit here in a particular example. So let's look at this example where I have one forward term. And so the equations look like this, okay? And this equation actually has a solution, a closed form solution, which is nice for us. So we can look at this. And this has its maximum here, right? And I should say that one of the main things we want to describe is actually the speed at which the sequences propagate. So these peak times will basically tell us this is the information that we're looking for, okay? And the peak difference basically gives us the inverse speed. Okay, so this is equal to 1 over 1 minus A0 bar. Okay, and this is just to show that this is comparing the linear system, which is the dashed lines, with the full nonlinear system. So this is the peak times, these T mu's, and this right here is that G function. So what you'll see is that the nonlinear G function, which is given by the solid line, so that corresponds with the full nonlinear dynamics, typically approaches this value that we predict. This is the edge of stability value, unless the edge of stability value is too high. In which case, basically, the nonlinear network goes towards it, but it's stopped because G of T is just fundamentally bounded for reasons that I haven't told you. Unfortunately, I don't have time, but just, that's the way it is. So it's bounded. And that means that this particular example decays to 0. Okay, so these overlaps will decay to 0. Whereas these guys all continue along without decaying to 0, basically. So they look normal. Right, and so that also corresponds with this guy right here, which is basically the failure. Everything else looks nice, but this guy kind of messes up. Right, and so G infinity therefore determines the stability of the system as well as noise robustness, it turns out, because when I add noise, remember I have this noise factor here, this will actually change this bound. So noise will actually push this down. So I need to make sure that whatever, this dashed line is, that is gonna be below that bound. And basically this dashed line goes down as the sum of the coefficients goes up, right? So this G infinity gives you noise robustness as well. And so what this actually means is that you can keep the sequence speed fixed while you change the robustness to noise of the system. This is probably easiest to see in this particular example because you see that the speed here, so I can set A1 equal to 0, or sorry, yeah, I can set A0 equal to 0. So this is A0 bar, so that's a little bit different. But if I set A0 equal to 0, then the speed will stay the same, but I can change A1 arbitrarily. And A1 basically gives you the how fast you go to the next, or it gives you how robust you are to noise and how far away from the stability edge you are. But A0, if A0 is 0, then changing A1 doesn't change the speed of the sequences. Okay. All right. So yeah, did I hear a question or? Okay. So let's continue. So now let's talk about the general case. So that was a special example. So I just want to run through kind of the basics of how we compute this because I think it's kind of interesting and potentially useful. So this is just like math minute here. So feel free to unplug if you're not interested. So the first thing we do is we assume that these equations are not periodic in mu. So that's the first thing we're going to do. We're going to say, okay. Oh, and I should say, in general, it's not easy you can't solve for this explicitly. So let's get an approximation. So we assume that's periodic in mu. Now, if it's periodic in mu, what this means is that this linear ordinary system of ordinary differential equations has a coefficient matrix, which is basically circulant. And that means you can diagonalize the system, right? Because circulant matrices have eigenvalues and eigenvectors that we know. That's a very nice trick. You change your system to periodic, and it's much easier to deal with usually. So this looks something like this. So this is like basically the solution to a linear ODE, system of ODEs, after you've diagonalized it. Basically, it's e to the eigenvalues. Here are the eigenvectors. And usually you also have eigenvectors over here. But because of the initial condition being so simple, it basically just kills that eigenvector. And so that just gives you a scaling factor. But so this is what it winds up looking like with this initial condition. And so if you take p to infinity and write this out as an integral, then it looks like this, okay? So basically all that you really need to realize about this is that it has cosines, it has sines, that's due to the periodic boundary conditions, and we want to evaluate this. Now, this integral, there are multiple ways of going about possibly analyzing this or computing an approximation to it. Actually, what I'm trying to do right now is to actually use a saddle point approximation. But the first thing that's not done. So the first thing that I did, which also gives good results, is a more naive kind of approach where you just expand the cosine and the sine terms here in the exponent out Taylor expansion to the first two terms. And you can see that cosine will give you like an s squared term, sine will give you like an s term. So basically you're going to have an s squared, an s, and an s term. So this thing's going to look kind of like a Gaussian integral. And the way to evaluate these kind of Gaussian integral guys is by using a Hubbard-Stratinovich identity, which, if you know it, you know it. If you don't, probably not worth going into. But, that's how you do it. Now, to write down the final solution more easily, we can define these things here, alpha and beta. And then we finally get this approximation, q mu of t equals this guy, where x equals mu minus t alpha and y equals t beta. So notice that we do get some of the basic behavior that we want, namely that the variance here is scaling with t. So as t gets bigger, these things will spread out. And the center of the bumps is governed by mu. So mu is kind of given us it appears into like the mean of this sort of Gaussian type guy. Of course, this y here makes it not actually Gaussian, but it's kind of looks pseudo-Gaussian. Okay. And so you can now compute the peaks of this thing. You can take the derivative set equal to 0, and you get that t mu equals mu over alpha minus beta over t alpha squared. Okay, fortunately, this part doesn't really matter because what you're interested in is t mu plus 1 minus t mu. So this part just goes away, and you're left with 1 over alpha. So that's like the actual governing the speed of the sequence dynamics is 1 over alpha. Okay, so if you don't like math, you can wake up now. And okay, there's still some math, but at least we're not going to drive anything. So this is just an example of what this approximation gives you in this case where we had this kind of scenario. So if you expand it out, it looks like this. Okay, so this is what the basically sequence speed looks like. And here, this is plotting the t mu's, the peak times. And basically, I just want to show that the actual simulations of the full network, the linear approximation, and this approximation given by all that gobbledygook I just went through, all lie on top of each other quite nicely. So this approximation is pretty good in practice. Okay, so I messed up my transitions a little bit here. Ignore these guys right here. They're just, they'll make sense later. Okay, so I have this guy equals, right, this is what we basically found out. And now I can use this to evaluate the particular case where I have exponential kernels. They're like double-sided. So that actually looks like this, right? So we have these double-sided exponential kernels. They have this form. And this is a common model in biology. And so here you can compute these coefficients ak and a minus k explicitly. And this leads to this expression here for the peak difference, which is the inverse speed. And you can see in particular that it is linearly dependent on this teak c. This teak c is basically that's the width here of these intervals. So that's how fast your tutor signal is changing, right? So we'd really like to know, how does the time scale of the tutor signal relate to the time scale of the actual network dynamics? And it's not one-to-one, but it is linear. But then these guys, so the actual shape of your kernel can modulate how fast your sequence dynamics actually go, depending on some tutor. So for instance, you could maybe do something, actually I haven't tried this, but you can maybe do something like, I'm just running through an environment, and this is giving me patterns of a particular time scale, but then the time in the network could be really fast. So this could correspond to something called replay that we see, where basically these networks are recapitulating the sequential behavior, but way faster than you actually see it in the behavior. Okay. So let's go ahead and plot some stuff. So here's that linear relationship that I was talking about. I'm just changing teak c. Okay, these make sense now. These are the peak, the time differences. And right, so I got the full network simulation, which is the solid lines, the linear guy, which is the dashed lines, and then the approximation, which is the dotted lines. And the linear and the approximation are really close. So even though we didn't do the full saddle point approximation, really the thing that matters more is going from the nonlinear to the linear dynamics anyway. So that's that. So I keep on pushing my cursor button. Okay, I did the same thing on this slide. So what happens when the tutor signal intervals are inhomogeneous, right? So this was all for fixed Tudor signal intervals. So let's say I have them coming in slowly, then they come in quickly, then they come in slowly again. So in this case, it looks something like this. So these are the time points. So they will, you'll go at a certain speed. This is actually speeding up. You have to think about it, but this is actually speeding up. And then this will slow down again. Okay. So it does work kind of, but if we look at the actual, this is the actual speeds basically, then we see, okay, we start at a particular speed. There's this weird speeding up that happens before we enter this, this weird slowing down that happens before we enter this faster interval. Then we go faster, and then we slowly, so basically outside of this is where the interval happens. So these dashed lines are the transition points where we go from one interval size to another. And then, so that's at 30. So then we slowly recover the original slow speed over this interval. So basically, I just want to say that this kind of sucks. It really doesn't do a good job. So there's something missing in this model that would, basically make this work a lot better. Okay, so I just wanted to throw that out there. And there is some work, exploring this, which actually it'd be good to have some papers up here, but okay, anyway. And if we look at the actual overlaps, then you see that, this is one interval where the distances look like this, then they go close together, then they spread out again. So we can see it looks a little bit messy. So, okay, let's fix that basically. Okay, to summarize, the ability to consolidate behavioral control into, automatic sequential action is thought to be important for animal behavior. Okay. And this ability may also be someday be important in artificial intelligence as we kind of try to do more naturalistic style things, like being able to learn a tennis serve in a reasonable amount of time, at least get reasonably good in a reasonable amount of time. Still takes a while to perfect it. That would be like naturalistic intelligence. Okay. Now I presented here a general theory of sequence memorization via Hebbian reinforcement of Tudor signals. And this theory predicts, sequence speed, and also shows how noise robustness can be modulated while keeping the speed fixed, or vice versa. So basically, you can modulate these two things independently. And this theory also shows how sequence speed with exponential kernels depends on the parameters of the kernel and, the way the Tudor signal comes in. In particular, the peak differences scale linearly with the Tudor signal speed. Okay. Just some quick future directions. At the beginning, I sort of talked a little bit about these discrete states and how that's kind of annoying, if you will. And also we assume that those states are independent of each other. But really, the neurons in the brain should be correlated, right? And perhaps we don't want to use this discrete state. So you could think of generalizing this model to a continuum of correlated patterns. That would be cool to do. Right, so there we go. This is this discrete model. Can we make that more of a continuum? And also, can we integrate like a memory module with other mechanisms, such as incremental refinement, on the general purpose controller to create, something that can do more than just memorize, basically. And with that, I guess I will finish and this is my boss. Thank you, Cengiz. We just worked on this together. And thanks to the Swartz Foundation, who really wants me to say thanks. So, thank you. Any questions? Yeah. Oh, yeah. So, it's a little bit of an all or nothing thing in that in the model, you are either going to decay to zero eventually, or you are going to persist forever. So in terms of length, right? Now, the derivation assumes that the number of neurons is much larger than the number of patterns. So if you wanted to start talking about capacity, like have P over N some fixed ratio, I'm not sure. Yeah. That would be interesting to look at. Yeah, I don't even really remember what the state of the results are actually in the literature. I'd assume there's some work on that, but I don't. So there is a paper that I'm familiar with. Yeah, actually, I don't think that is known. It is known if you take more than one sequence right? And so you have multiple sequences basically. And then there's like, okay, kind of more capacity arguments for these different sequences. That's easier because one sequence is independent of the other kind of dynamically and everything. But when you actually have a sequence, it's a little harder. It's a little trickier, I think, to compute capacity. I would imagine it's probably similar to just Hopfield style where you have these patterns, but who knows? So some fraction of n. Any other questions? Yeah.
I'll find it later, but I'm curious to the extent to which the feedback signal could be much sparser or something like that. But here, if I understand correctly, it's kind of the first time step.
Yeah.
Bird analogy, right? Like the father bird is young, pecking the berries.
Yeah.
For instance, like a complex environment. And then you can forward. Do you see extension between your work and that type of context, or is it?
Yeah. So that's a good question. I do kind of see myself as cheating. I think in a good way in that, in that I consider this to be this part of the problem, which is I already have something that can deal with the problem of, sparse rewards, that can deal with the difficulties of learning. And all I want to do is I want to take what I'm doing and, okay, I maybe want to modulate my memory storage with some sort of reward signal, but I'm basically just storing things, and I'm not really using reward signals. I'm kind of letting this stuff take care of that. So the tutor signal itself is in some sense taking care of that. But yeah, how to do that is another matter.